Now let us know a brief introduction about SABBIT, what it is and what it does. So, Students Association of Biotechnology, Bengal Share Technology, or more commonly known as SABBIT, is the biotechnology club of Bengal Share Technology. It was created by our seniors under the guidance of our biotech teachers, mainly Dr. Saurav Kanti Sarkar sir. Well, it is the first time that we are organizing an intra-college event under SABBIT. Every year we organize various seminars and workshops with various reputed national and international scholars. Being a part of our society helps the students gain insights on biological R&D and make them realize the value of biological approach in various industries. We at SABIT always value knowledge beyond our syllabus and support the union and teamwork of students from several departments into solving one common goal. Due to the modern diversification of technology, engineering is not restricted to only one area. Hence, we value our members to know cutting edge interdisciplinary research that are happening right now in various places of the world and try to connect with people who are doing that themselves. Thank you.
Thank you so much, Nikita. Now, we would like to start the journey by the ceremony of lighting of the holy lamp by quoting Lord Buddha, "If you light a lamp for someone, it will also lighten your path." Since it is in an online platform, we are doing that virtually. Now, I would request our respected. I'm sorry, my video was off. So, now, I would request our respected principal, sir, Dr. N.C. Ghosh, to give the welcome address to everyone present here. Over to you, sir.
Yeah, thank you very much, Vidhiman. I, on behalf of Bengal Institute of Technology and on my own behalf, welcome all the participants, guests, and also the faculties, HOD, Head of the Department of Biotechnology, for attending this 2 days symposium on cancer biology. So specifically, I welcome our guest speakers, Dr. Gazala Sultan and Dr. Inamul Hassan Mother. They are our guest speaker for this 2 days workshop or rather symposium. I also welcome Head of the Department of Biotechnology, Dr. Bosumita Rai Chaudhuri. So I also welcome the faculties of biotechnology of BIT. And I specifically welcome Nikita Agarwal, who is the president of SABBIT. SABBIT has said it is the Student Association of Biotechnology of BIT. And also Mr. Riddhiman Mukherjee, who is facilitating the today this activity today as an anchor. And also my beloved students and participants who will be participating in these 2 days events with some specific objectives. So this I'm glad to know that it is 12th annual symposium. Means before that there were 11th annual symposium, and now it is 12th annual symposium successfully organizing by our students. And you know, Miss, when you organize any symposium or any Miss debate, poster presentation, which maybe which you do generally physically, and also nowadays it is virtually. These actually aspire all our students. Means how do you fight with your say friends, or how do you groom yourself, right? So these events, all these events are very encouraging all the time. So uh, in coming 2 days, I understand all our students and also our guest speakers, they will deal with the total activities nicely, and I'm happy to just share that when I saw that, okay, they have organized say multitasking activities like say debate and also poster presentation. So this will be there online. So as a results, they have got some challenges, So during this COVID-19 pandemic, we all are physically disconnected, and our aspiring mind setup are also almost blocked. So we have to come out from this situation very soon. We will come out from this situation very soon, and we will fight back and win from the COVID challenges. That we believe and understand. Will come out from these challenges. So what I understand is organizing something digitally is a challenging task. When, as I said, when it is a multitasking activities. So our students have taken these challenges to organize this symposium by having say debate as well as poster presentation. So uh, uh, we understand that, okay, our students and also our participants will do it very nicely in coming 2 days, and I welcome everybody, and I also I convey my sincere thanks to everyone for organizing this in coming 2 days, and I hope that it will be a successful event. And if there are anything required from my side, so please let me know. So today I may not be able to stay for a long time, so I will be excused for that. But tomorrow and also later stages, I will be available for at the services for my students and also for the department. With these words, I once again welcome everybody. Thank you very much.
Thank you so much, sir, for your kind words and inspiration. Now, we shall end this inauguration ceremony and proceed with the technical session of the day. It is our pleasure to welcome two great academicians and researchers, Ms. Ghazala Sultan and Dr. Inamul Hassan Madar, who has kindly accepted to be here as guest speakers and also shared their knowledge and expertise in the domain of cancer biology. The entire team of SABBIT heartily welcomes you in our symposium on cancer biology. Firstly, our morning session would be divided in two parts. The first part would be spoken by Ms. Sultan, and then the second part would be continued by Dr. Inam. Starting the session, I would like to introduce our first speaker to you all, Ms. Ghazala Sultan. Ms. Ghazala Sultan is a senior research fellow at Aligarh Muslim University. Her core area of expertise lies in clinical cancer genomics, specialized in breast cancer biomarker discovery. She graduated BCA and MCA from the prestigious AMU. Ms. Ghazala has been guiding for major to minor research projects for bachelor's, master's, PhDs, and working professionals from AIMS, Apollo, and Manipal University in collaboration with BioCuration Bangalore. Her areas of specialization are RNA sequencing, exome sequencing, microarray analysis, mutation analysis, junction and fusion-based study from clinical cancer data. Ms. Ghazala has anchored more than has authored. Sorry, Ms. Ghazala has authored more than 8 international research articles in high-impact scientific journals, including LWT, Molecules, Bioinformatician, and has registered 6 inventory patents under Intellectual Property of India. She has also worked on nearly 10 books to her credit. Additionally, Ms. Ghazala is a lead member in management team of Edomics, Theragnosis Lab, and Sana Hospital and Healthcare. We at SABBIT heartily invite you in this symposium and wishing that the participants would learn a lot from you. Over to you, ma'am. Hello, ma'am.
Yeah, hello everyone. Yeah, Ghazala Sultan this side. Let me just share my slides so that we can start it. Hello, am I audible to you?
Yes, ma'am.
Okay.
You're audible, but actually, I can't see your presentation. Actually,
My slides are not visible.
Yeah, it is visible.
okay, then it is my problem. Sorry,
Okay.
ma'am.
So is it visible to everyone?
Yeah, it's visible, Gazala.
Okay. Okay. So hello everyone. This is Ghazala Sultan, and I'm pursuing my research in cancer genomics from Aligarh Muslim University.
Could you make a full slide? Because I think like you're changing the monitor.
I think it's a full slide only,
No, no.
not
Check it
full
out.
screen.
Change the screen which you're sharing.
Okay, sir. Now is it visible? Now it's a full screen, I think.
Yeah, yeah,
Yes,
it's
ma'am.
fine right now. Yeah.
Okay. So, okay, so I have introduced myself already. So we'll start I would like to start the presentation with this quote. Find something you are passionate about and keep tremendously interested in it. So, this quote has been my source of motivation since the time I decided to pursue my career in cancer research. And I hope this will keep you motivated as well. So, let's start the presentation for today. So, we will go through the cancer prospects in career prospects in cancer biology after completing the degree course, whichever you are pursuing, and this will be definitely helpful for the undergraduate and master students and everyone whoever is interested in cancer biology. So, whenever we talk about cancer biology, the first question arises that what is cancer? So, and it is not a new question or new term that we are not, anyone is not aware about. So, we all know that cancer is one of the leading cause of death in India and over the world as well. So, the word cancer instills more, we all know that the word cancer instills more fear than death. Whenever we hear the word someone is having cancer, it definitely it comes in mind directly that it is something related to death only. That's how people are so fearful with this particular disease. This disease has become a big threat globally, irrespective of social status, gender, or physical fitness. So there could be many different reasons for cancer, and it is not definite that these are specific 4 to 5 reasons that could lead to the cancer. So there are multiple reasons, and some of the reasons could be like can be concluded in two parts, like internal reasons, for example genetic functions, mutations, immune conditions, and then some of the external or environmental factors include food habits and pollution and others. So basically it is a heterogeneous disease which have different phases, different forms, different molecular signatures, and different forms. So it is not definite to one particular definition that someone is defining with this particular definition as a cancer, and then that's all for cancer. So we cannot define in few words. It's a broad term. So let's just move towards what are the different career opportunities in cancer. So basically, we all know that cancer is one of the most researched and most studied type of disease all over the world. Like various researchers and scientists are involved in finding the best possible solution to treat the cancer patients. And we can see as statistics is there that over 33 lakhs patients are reported at any given time. Annually we have lakhs of cases and deaths as well. So we can see a noticeable stats that 21% increase in the number of cancer patients between the year 2004 to 2020. So over the 16 years, 21% of total cancer cases has been increased. And also the most disturbing part is 71% of deaths occur in this in the age that is the we can say the middle age, the 30 years to 69 years of age group. So that is one of the thing that is that we are more concerned about, and all the research are going on how to treat it, like fully treat the patients whoever are like cancer prone or who are getting this particular disease. So why is cancer research so important? We all know that how important or how important our lives are, right? So if we are going for in the field of cancer research or cancer biology, then we can save lives and we can give a better life to the people who are struggling with this particular dreadful disease. So that's how where it is very important to go into this field, to opt for this disease and go forward, choose a career that can that can help people and save lives. So, yeah. So, before going forward and choose a career, for example, you just completed your degree and you have found that there is a vacancy at particular position, which is something related to the cancer skills. So that is not enough that you are going to apply for that particular position. You should know that where your interest lies in, where you are interested in, what you are passionate about. Like because in cancer, it's in cancer research or cancer biology itself, if you have the knowledge of cancer biology, then there are different within the same particular area of the study, there are different career opportunities. So you should know that how and what in what particular things you are interested in. So there are few questions that we all need, whoever are like interested in pursuing their career in cancer research or cancer biology, whoever have the skill sets related to that. So we have to first ask few questions to ourselves. because we may tell something different things to different people, that okay, I like this thing and other things, but we cannot lie to ourselves, right? We know perfectly that what we want actually. So the first question here comes is, do you enjoy making a difference in people lives? So we all know that, like, it is not a new thing that we don't know how we can make difference in people lives while we are pursuing the cancer related any career. Next is, do you enjoy solving problems and challenges? Because once you are into the career, there will be several problems coming up too. Then you have to solve with that. You have to solve. There will be uh, like puzzles. You will feel like puzzled, like you are not able to solve the question, or it is too challenging to come across and to help out. So there will be different questions. So person should be interested in solving problems and taking up the challenges. So that will be one of the plus point for the students or who wants to pursue the career in this field. So we all know that what kind of problem solving problems could arise if we are opting this career. Next is, do you enjoy working as a team or working independently? Because that also depends that what type of career you are going to choose. For example, if you are going to join some research team, then there are research teams where they work in a team. There are researchers who work independently as well. So there are different, each career require different type of teamwork or independent work. So we need to be sure that what we need, where we are comfortable, or where we are ready to take the challenges. So next is, would you like to help people when they need their help the most? So we know we all know that how we can help the people. So like, nothing is more expensive than life, right? So we can help them in many ways. So that's how we need to be passionate about our profession, whichever we are going to choose. That's why it's important to that's why it is important to understand and first think or choose your career wisely, that where your interest lies in, so that accordingly we can proceed further. So consider the joining the team of fighting, like you will be joining the team of scientists and researchers, where they will be doing numerous of the researches based on the, like, finding the different problems, mutation types, gene expression, how they are different from the normal people to the cancer samples. Like, we have been doing analysis using the real time data of the cancer patients and how the genes are different. Like, different kind of studies are there, dry lab, wet lab studies are there. So wherever, whichever type of things you are interested in, that will definitely help to choose your career. Okay, so the problem, what is the major problem is, so that which is not negligible as well, that out of two women, there is one man that will definitely in their whole life, will one man will have definitely get the cancer, and out of three women. So that is the ratio how it's going on, and this is kind of a current ratio that I am providing here. So that is what one of the major problem and facts based on the survey. This is the latest survey where we have identified that 75% of people are there who have known the cancer which is running in the family. They have either the relatives or the family member who have been diagnosed with the cancer, and total of 88% people know someone with cancer. Even 75% have in the family, but remaining know that someone is there, a friend is there, a neighbor is there who got cancer, like that. So this statistic we cannot neglect, right? It's quite huge statistics, and we should be concerned about the lives of people who are our, like, fellow people, colleagues, and other people as well. So yeah, so this is one of the reason that one of the greatest problem that we need to keep in mind and should choose the career so that it will make add some meaning to the others life as well. Next, with the along with the problems, there is a good news as well that up to 70% of all cancers are preventable. So there are different preventive measures that we can take and in the proper way, and we can prevent cancer. And there are treatments available as well now. So yeah, so there are good things as well along with the problems. And as of now, currently there are over 10 million cancer survivors today. So that is because we are progressing into cancer research. That all the career people whoever are in this career, so they are adding meaning to the life, they are saving the life, and there are over 10 million. So this is quite a big number where 10 million people are a cancer survivor today. So yeah, there is quite a great scope in this field, and you we should go forward and choose the career. So yeah, would you like to have your career many meaningful jobs? Like there are a ton of jobs which are available. Whoever the people who are having the cancer related skills. So like even they are from bioinformatics, even they are from biotechnology. So biotechnology people will be having the wet lab analysis and those type of skills. So every so whatever skills you have or you can learn, even if you are not from that kind of background, so you can learn and then you can go for, if you have a passion, if you have an interest in that particular zone, so you can learn and go forward for this career as well. So that is not one of the major necessity that you should have a degree from that course. You should learn the things, whatever degree you have, you should learn the things. Once you have the skills, you can opt a job and then you can go forward for the career. Then there are yeah, then there are deep personal satisfaction. We know how, like how it feels when we help others. So like we know how we can add the meaning to the people's life if we are choosing that career and treating someone or helping someone in any way. So that will be one of the best thing. Then a variety of career choices are there. For example, there are different jobs, different job opportunities coming every day, even every hour we can say that frequently job opportunities are coming in this field. And also, if you are not satisfied or if you are getting better job opportunities which you are having currently, then you can also skip to your current job and you can move to the next job which is having better opportunities and you think better scopes are there. So there is a limitless growth in this field, and you just need the skills and few basic requirements which are there for it, that is respected to each job or the career path whichever you are choosing. So that will discuss definitely. Yeah, next is economic consideration. So yeah, definitely, it is just a common thing. Like we, no matter how usually we say that money does not matter, but it does matter, right? We can help to people, we can help ourselves, we can uh, help someone to fulfill their basic necessities of life as well. So that matters. So that's why we give so much effort in this particular field or any particular field whichever we are studying. We put our all the efforts and study, go forward, learn new skills, and then get new job opportunities to be eligible for new jobs which are coming with the more bigger packages, so that we can broaden our skill sets as well. We can help peoples, we can do many different things if we have. So we cannot neglect this section as well. And there is quite a good figure where you will be eligible to get once you have the career in this particular field, whichever career you are choosing, either researcher, either social health worker, or other career as well. So let's see. Yeah, so hope you guys are hope I am not too fast. So if I am too fast, you can tell me. We can go slow as well. So is that fine, or I am going too fast?
Yes, ma'am,
Yes,
it's
ma'am, it is.
fine.
It is okay, ma'am.
It is fine.
Okay. Okay, that's good. So if anyone have any query, you can just ask me, or later of the session also we will be having the query sessions, so you can also ask them. So yeah, next is the opportunity. So what opportunities are related to what things you might be having the questions related to this. So we can get the career in prevention, control, detection. So there are different type of methods, like how to detect how to detect cancer if someone is having cancer. So for that there is a different area or different career path, so that where you can be a researcher, you can be a physician. So that kind of things are there, how we can detect. So you can just see like what mammography is there to detect the cancer, blood test, and we do genetic testing for someone who is having cancer, to go for like to see how their genes are functioning in the cancer samples, and how it is different from their normal samples. So like that, those kind of things are quite insightful. And if someone is interested, if you are interested, and you should be interested, because since you are attending the webinar, it definitely means that somewhere you are interested to know and to pursue the career. So we should be aware about all the aspects of this career, so that accordingly we can see, like someone is really good at mathematics, they can they can do mathematical modeling and molecular sciences and other things. So there are numerous of the opportunities. Actually just wanted to convey that one. Then opportunities in treatment, then how we can treat the patient. So there are different options like surgery and chemotherapy. Emotional counseling is also there. Not only that you are going to be a physician, or you can opt the counseling option, that where you will be counseling the cancer patients for their emotional healing, because we know how someone like will be there if someone found that they have the cancer suddenly. So yeah, there are various options we can see. So what you will work, apart from the like direct contact of the patients, like patient will be there, so you will be treating the patients, or you will be suggesting, like once doctors are treating the patients and you see the report, you can suggest better options, better medications, if you have if you have the knowledge in that field. So that is one of the, you can be an educator. If you have the knowledge, you can educate people, you can teach the fellow people or the students who are eager
মোহাম্মদ
to
ভাই
learn, who
যেটা
wants to
টেক্সটটা
learn what
করেছিল
you
পিন
know
করেছে
already,
ওটা
because you are
এখানে পাঠা,
experienced in
তুই
that one.
যেটা
So
করেছিস
yeah,
ওটাও
there are
এখানে
different
পাঠা, আমি
opportunities as well
টোটালটা একসাথে পিন
where
করব,
you can
বুঝতে
work
পারছিস?
on. Next is where you will work. So yeah, there are different options to work as well, but in terms of laboratory, we'll be working as a research scientist, oncologic pathologist, medical technologist. So yeah, different based on the skill sets, what is skill. So yeah, here again I want to mention that skill sets are really important, what skills you have, based on, because every time you go for any applying for any job, they will be mentioning that what skills they need, what you will be doing in their lab or wherever you are applying in that particular job. So again, one of the most important thing is skill. So you should be more eager to learn the things, because every day by day new technologies are coming, new methods are coming up for treatment, detection, and cancer detection and everything. So we should be always be hungry to learn new things, whatever is coming up your way or you are going through. For example, let me just give you example that you are applying for some, or if you will be applying like after this, or after an year, if you are applying for a job and they are asking for this skills, we need the skill that where you can do genetic testing, where you can do this type of data analysis. So you should know that, okay, let's say that you don't have that skill sets, so what you will do, you will not apply, right? Because you are not having the skill of like that skills which they require. So what you need to do, but you are very much interested in cancer research, or so the whatever information or the knowledge you have from your degree course, so you have the knowledge for cancer biology, right? But the job which is asking for some other skill sets which you don't know, so you should be always hungry to learn the things. If you are interested in pursuing the career in this field, you should try to note down those skills, that okay, this job I am interested in, but the skills I don't know. So you should note down the skills and try to learn those skills. There are many sources to learn the skills. So now there are many sources on the web. We cannot say that there are very something not available on the web now. So yeah, flood of data is there. So yeah, so you should learn the things if there is not, if you don't know something. So yeah, with whom you will work. So yeah, of course, depends on what career you are choosing, because there are different options. So accordingly, we will be doing work with like NGOs or scientists, researchers, whichever career you are choosing, that will go with that. Then next is, okay, what type of careers are there now? Then career, consider a career in medicine. So there are like every career have its own benefit, because after all you have gained the skill, right? So you, there will be the benefit that how you can save a life by being a physician, how you can like add meaning to the life, treat the patients, do research, education. There will be continuous learning, because every time you are treating any patient or any new patient is coming, there will be, because cancer is a heterogeneous disease, so there will be different kind of case. Not like, for example, someone is having a stomach cancer, so the next patient which is coming with a stomach cancer itself will not be getting the same treatment which you are giving the for the first patient, because same stomach cancer is not, every patient have their different variability, or different kind of variations are there in each cancer patients. So for that, there will be a continuous learning process. You have to go through that, what type of variation is there in the next patient, for by genetic testing and all the things. And then you can understand that, okay, this is the new kind of variation is there in my new patient, and I should learn about this mutation and or this variation, what research has been done, what I can prescribe medicine for this patient. So there will be continuous learning, no so, and we should always try to learn at every stage of life, because learning never stops. So yeah, we should be just passionate and enthusiastic about learning. Next is, okay, so what kind of educational requirements it need to be a physician? So yeah, so that is obvious degrees, which are bachelor's and master's degrees. Then doctorate, that is the PhD degree in medicine, or and the other like experience, some experience they require. But you, if you have the basic degrees along with less experience as well, so that is totally fine. That depends on like opportunities, job opportunities, to that varies with the job opportunities. For example, in some institute they ask for less number of, even if you are having minimum of 2 years experience, they can have you if you have the skills, if you do well in the interviews, because in interviews they will judge your skills by how you speak, what you speak. So that's it. And what all we can do in being a physician. So yeah, in oncology we can do in radiation oncology. So oncology is a field of like a study of the cancer. So there we can do different kind of research, public health, biomedical engineering, information technology. We can do different kind of work being of a physician. So yeah. Then there is different like options, like what are the things we need to do. We can help in prevention and control, we can help in detection and treatment, and other things whichever are involved, where you will be needed, where you can work. You can work in hospitals, medical schools, then cancer center. So there are, according based on because you are a physician, so you of course you will be involved in treating the cancer patients. So of course these kind of centers you will be needed, where you will be working. So you need to be prepared, be skilled in that way, so that you can work in those environments. You should have enough skills so that you can because you will be having the responsibility of a life, right? Someone's life is in your hands once you are a physician. So you should be very careful that you have the enough skills, you are prescribing the right things. That's all. Yeah. Next is yeah, so there are you can do specialist like there are oncologists. So general oncologists are there who treat who can take any type of cancer patients, and they can go through them, they can try to treat and detect and treat the patients. And there are you can do also specialties in different types of cancer as well. For example, lung cancer, stomach cancer. So for that also you can be specialized in different cancer. And then in oncology study there are different types of like career options as well, like medical oncology you are interested in, or radiation oncology. So that those are also different career paths which you can choose, where you can specialize on certain type of oncology, and that will make you a specialist, where you can be a senior consultant for other fellow oncologists, or the any kind of patient if coming up with some complexities in like cancer treatment. So yeah. Next, you will be having an option to consider a career in public health, where again everything is having its pros. So yeah, you can help in identifying health problems. You can help in giving suggestions, but for early detection, how people should be, how what measures particularly they should take to prevent cancer. First, at first place, we should try to prevent cancer by any kind of factors that we need. For example, food habits or environmental factors, any kind of thing, whatever possible, we should try to live a healthy life to prevent any kind of disease, not only cancer. Then what particular measures we can take for early detection, and then other health measures that can help in avoiding cancer. So that could help. And they can also being a public health scientist, you can also suggest that which oncologist is specialized in which particular cancer type, so they can refer you to the concerned oncologist. So like that, that's how you can help people. And that will be really like helpful for people who don't know, who have no idea, who have no people who can suggest, give better suggestions. So you are a source, you can help that way as well. Okay, so educational, so yeah, basic degrees we need, that we all know. Even if without the basic degrees, there are we have seen there are like agencies, there are companies who are also there, who just grab the people who are interested, and they teach them, they uh, provide the skill sets to them, they try to teach them, and then they hire them for the particular jobs that they have the requirement. So that's also fine, but we should because we all have, like you, everyone is having the degrees, right? And you are in the college, so yeah, it's you have the more chances to get on that position rather than the people who are not having the degrees. So yeah, this is one of the basic needs that is there. Yeah, so basic needs, we all know that bachelor's and master's is there. and based on the which position, for example in public health officer as well, which position, on which position you are going to get hired in this particular job, then based on that you need, sometimes they hire masters students as well, masters pass out. So sometimes if you are going on a public health scientist, so you need being a PhD, and then you should have some experience as well. So based on, we know that how it works when we go for like when go for job. So based on the position level of the job, or what position you are going to be get hired, so they will be asking for that particular type of educational requirements, so that we need to fulfill. Next is yeah. So and again, we I said how, like for example, if you don't have the experience, you can opt for next job, or you can just you can write it, mail it to them, that I have these particular skill sets, I have this number of experience, but I am not fulfilling on that particular, like that number of experience years which is mentioned in the job. So there might be possibility that you will get the job once you are done with the interview, one-to-one interview that they get. So yeah, there are different options. You, it's just required that you need it and how bad you need it, so everything will be possible. Next is considering career in research. So yeah, we all know that how like what is the scope in cancer research. Doing research is of course one of the major thing that we need, because once research is done, then only we get into the conclusion that what kind of disease is it. We can go to the molecular level, we can study the molecular level of the disease, we can understand that, and then we can make find new relations based on the previous studies which has been already done. So yeah, so those are, this is also is one of the major like career option that we should choose. And then we can improve, like we can implement our skill sets, because everyone have different abilities of learning, of generating the results, of presenting your thoughts. You have different ideas, the next person have different ideas, and when different people with different ideas work together they generate a new idea, and that is what we call a novel discovery, a novel finding. So that's how it works, and that's how research works. That's why most of the time research works are in team, so that there will be different people and there will be a new finding coming up, which is required for the any kind of research that we are doing, or whichever is targeted for the particular. For example, one gastric cancer patient is there, and it we have tried every way possible, whatever treatments are there we have tried. Chemotherapy is done, radiotherapy is done, but it is not getting treated. The cancer is getting worse and worse day by day. We are not able to treat that. So in that case, we need the sample, we need to study that what kind of variations are there in this particular sample, why this tumor is not being treated with the other method that we have been treated so far for the because cancer varies from people to people. It different types where its type varies from people to people. So that's why when we find something new where there, for which there is no existing solution, so for that we need to research and find novel thing which has not been found yet, and find which generates a new knowledge and add into the research discoveries. So that's how research is important, and which is also required. So yeah, there is a great scope in research as well. And education, then again requirements that is needed, so that we all know. And then after bachelor's and master's, so we to do research, we have different options. Even you are from biology or genetics or any biotech or anything, or your PhD is specialized in any field, so you can go for research. Every field is having research now. So yeah, whichever you are interested in, you can go. Because I am from computer science background, because my bachelor's and master's was in computer science. Now I am doing cancer genomics. So yeah, everyone can do. It's not necessary that you should have some uh, background from cancer biology. You should have all the thing. But if you already have the skills which you are going to pursue in your further career, so that will be one positive plus point for your career, and that will help you definitely in your further research work or whatever career you are going to opt. So yeah, that is really good. Yeah, so what we discussed that based on the research programs, we need to take any specific type of research type, so that we can research on that specific. Because when we go for research, so we opt any specific type of cancer that is more targeted study, and there then only, because every cancer is different and having different variations, and even within the same cancer there is different variations. That depends on person to person as well. So we know how much diverse cancer itself is due to its heterogeneity. So it is preferable that whenever you are going into career or in research career, you should opt any one type of cancer. Just go into it, dive down in it, research everything possible, whatever data you are getting, whatever from wherever you are getting the data, just get into it. No, try to find everything possible inside that. Go do insightful research, and then accordingly you can be a specialist in that particular kind of a cancer, and that will be more helpful rather than going for every type of cancer. And yeah, and just having the overview knowledge of all type of cancer. I will suggest to have like opt for a specific type of cancer, and just do a thorough research based on that. Next is, yeah, field of discovery. So new knowledge for the cancer. So what kind of new knowledge we will be generating? Because definitely, if you are going into the research background or research study, so there we will be of doing research. So we are research always aims to the new discovery, because everything is already, whatever has been researched is already published, and we know. So with the help of those information, we should go forward and find the new information that has not yet been known. So yeah, there is different opportunities for the discovery in when you go for cancer research, or even any type of disease. It's not specific to cancer as well. We can propose new treatment procedures, because based on the whatever results you are finding, that how, when I give the example of different, no, that cancer vary from one stomach cancer patient to the another cancer patient. So one patient is getting one treatment, the second patient is not getting the same treatment. So we have to propose the new treatment procedures, right? So then we have the different scope in research field, because we found something new for our new patient by doing the research, whatever data, that sample that we got from their tumor. So in that case, we will propose new treatment procedure, that okay, this medicines will not be effective on my patient, so I have to opt for the other medicine, which will be which is having more probability of working on the new patient. So that's how we proceed for cancer treatment. Next is, okay, so there is a universal need of researchers in hospitals, cancer centers, everywhere, right? We have the knowledge, and we have the enough knowledge to treat a patient, save a life. So everywhere the researchers will be needed. So yeah, it's one of the best field, I can say. Yeah, you may find it biased because I am into the same field, into cancer research, but in general also can say that it is one of the best field if you are really interested in cancer biology and want to go more into it and learn about it, and yeah, help and add meaning to the people's life who are struggling with this particular diseases. Additional career paths is also there. You can be a dietitian, nutrition, so how, what kind of diets people should take to avoid this kind of like cancer, like how to be healthy and what kind of nutritious values should be there in their diets to stay healthy, to avoid cancer, like we avoid taking fats and like that. So that is just a general, but there are different options, career paths, apart from being researcher, apart from being physician, public health scientist. There are different options, what whichever we are interested in. Genetic counselor we can be, we can suggest people to go for which particular oncologist, which particular medicines, or like which particular treatment opportunities are there, treatment facilities are there. So those kind of, there are different career paths. Actually, these are very few which I have just listed, which just came up, like which are top few career paths, but there are too many. So that depends on, again, that depends on skill sets that you have. And yeah, so, and cancer knowledge we all need. If you are going into the if you have the going into cancer research or cancer based cancer biology or something, so we need the knowledge of cancer biology. So that's how it works. Next is frequently asked questions with the people who are into cancer research or in cancer field, is how to apply for the positions, even for PhD or jobs. So yeah, I will suggest everyone know that how to apply, right? Search for these jobs and go to the portal, just apply that. I will just point out here that try to go and apply from the official websites. Always try to go for official website, that will be having more authentic positions given there. So yeah, one of the thing is there. Then okay, how long is the PhD degree takes, because PhD degree is most of the times when we go for or opt for higher career path, career in cancer based research, then they need PhD degree. So it will minimum take 3 to 5 years. So yeah, even in 3 years also we can complete after the masters. So yeah, next is, which is most funded cancer research, that is the breast cancer. We can see here the 460 million dollar we can get if you are having some project in breast cancer. So that depends on actually objective of the project, and what project you are doing, what is the objective, how it will be helpful for the like what is the possible outcome that you have planned, what is the hypothesis you have made. So that will be scrutinized and go through several procedures where the experts will be there, and they will be going through your proposal, and then accordingly. But there are people, like prostate cancer is having the highest rate in the men's for cancer and for cancer deaths. But the most researched is the breast cancer, and that is in women's. So that's why because breast cancer is most researched, that's why it is most funded also. So, and followed by that there are other type of cancer also. So it does not mean that if breast cancer is having the most more funds, so you should go into breast cancer research only. That depends on proposal to proposal. Even if lung cancer is getting 200 million as in the funded projects. So, and if your proposal for breast cancer is not that too strong, you will not be getting that $200 million even for your projects. So that depends on proposal, what is your objective, research objective and all. Then which countries are leading in cancer research? So there is Finland, Australia. So maybe if you are checking looking for the jobs or careers in cancer research, so you can just go through the job opportunities in the following like countries where cancer research is leading. They are doing really well, great researches are coming out from their laboratories. So like that, even in India we are also doing great. But yeah, top 5 are from Finland, Australia, Netherlands, USA and Canada. So yeah, whenever you are going through research papers, like if you are interested already right now, right, so you know you need to go through research papers. So try to get or grab the research papers from these kind of laboratories who are doing like who are leading, and they are doing really good, they are famous, and they are producing more number of good quality papers and good journal papers like Cell, Nature. These kind of good journals are there. Then good laboratories which are from like Finland, Australia, for an example we can see, because they are leading in cancer research. So probably those who are leading of obviously they are doing good amount of research and with great results as well. So we should try to grab the research articles, research papers or their tools or something, so that we can see and we can also learn their standards, what is their standards of learning and understanding, producing the results then, and we can match and we can upgrade our standards as well, how the other countries are leading in this particular research. So yeah, that is one of the thing. Then future, how so future is like we all know that how it impact life of the people. And so future we can say that in terms of new drugs, we need to make new treatment aspects, new treatment technologies or strategies we need to make to treat different type of patients with different type of treatment or medicines or different doses we need to. For example, one patient is having the same dose, but the other patient cancer type is not working with the same dose. So we need to increase or decrease the dose based on the cancer type or what is the heterogeneity or changes in that particular type of cancer. So yeah, that depends on cancer to cancer. So there is a lot need to be researched or need to be done in cancer research. So yeah, it's still going, and there is of course a great scope in future as well. So yeah, Jandrew from Gene Tech in stated that this is one of the most exciting time. So he stated a few years back that this is one of the most exciting time in cancer research, and I believe that the next 10 years is going to be a golden age. So yeah, that quite motivates the people who are interested in cancer research or who are already there. And yeah, so all the best whoever are interested. Next is top 10 cancer specialty hospitals in India. So we are also doing great and providing treatment to the cancer patients who are coming across. So there are like AIMS Cancer Institute, Apollo is there, Gujarat Cancer Research Institute is there, and there are numerous, like there are top 10 I have just listed, but there are other cancer research institutes as well where like cancer patients has been treated. Yeah, job related companies are there. So these are kind of official websites I have given there, where you can just go to their official websites, check for the job opportunities, and you can check that where your skill set matches and you can apply there. Even if your skill set does not match, you should learn the skills. Okay, so any queries if you have regarding training and workshop related to cancer research projects you want to do, job consultancy, India or abroad, cancer patient consultancy. If you have like someone known with cancer and they need consultancy or the treatment, you can just consult with us. So yeah, given are the email IDs for Dr. Inam and mine. So anytime you can contact, or you can contact with Ridiman. He have the details of us. Next is thank you for listening. Will end up my presentation with this thing that work hard, have fun, make a difference. That's what worth it, right? Otherwise, just studying, um, passing the exams is not enough, right? We should have some mission to do, some make a difference to the life, add meaning to the other's life. Yeah. Thank you all. Thank you all. Any queries?
Thank you so
Um,
much, ma'am.
Welcome. Yeah, tell me.
So there are a few questions which has
Mm-hmm.
been generated. So
Yes.
first, the first question is there is a question. Cancer biology. You have mentioned in a slide there.
Yeah.
It was mentioned that cancer is happening in one in two men and one in three women. Can you
Yeah. Right.
please explain why it might be happening?
Okay, so there is no particular specific reasons that why cancer is happening, because it can happen to any next person. We are not sure that to whom it is happening. So there are various different reasons. There could be different reasons for its incidence rate. Like as I mentioned, there could be internal reasons like genetic factors, mutations are there, different, for example, or the food habits, environmental factors like what you eat, how you live, what is your health, living style. So that can matter there. So there is no specific thing. What I said is it is quite a diverse thing. When we said cancer, when we say a cancer term, it is a diverse thing. We cannot state it in one definition that this is cancer and this is why it happens. Not like that. So there could be many reasons.
All
Yeah.
right.
Yeah.
So there is another question. Can you
Mhm.
please explain the role of nanotechnology in treating cancer?
Okay, in nanotechnology, how we can treat cancer is like there are nano sensors. Okay, so that we when we for example, there is a cancer or tumor in the patient, and there are new nano sensors which carry drugs inside the body, or which they can go inside the body and they can destroy the cancer cells where they are acting, right? So that way we can help in destroying the cancer cells and get it back to the normal cells. So that's how one way we can treat and help the cancer patients with the nanotechnology stuff. And yeah, there are other ways also, so that need to be studied. Yeah.
All right.
Mhm.
So then some of us are interested in your research work. Can you please throw some light on that?
Okay. So my research work. So as like I have been introduced that I am from breast cancer research. So I am working on early detection of breast cancer, where I have involved I have a specific pipeline for my research where I have made I have been adding machine learning algorithms which can classify the normal cells to the tumor cells. For example, when we separate the cells from any human, so there are maybe on the slides we get the normal cells and also the tumor cells. So how my algorithm will classify the normal cells and the tumor cells in different categories, and then how it will classify that how the when the tumor is extracted, then which stage of cancer is that of when we are extracting from the patient. So then when we get the samples, it will also classify the tumor stages. According to that, we will be trying to get the samples from the early early cancer stage, where the tumor is just in cancer stage, when we just see the swollen up thing or the lump, something like that. So that how it starts in breast cancer. So when there is a lump found, so at that particular stage also we get the samples from the patients, and we study that what is the variation from the normal tissue, then the lump tissue whenever it is falling, or is it next by following up the patient after months, like is it growing into cancer. Okay, if this for example, one patient I am following up from the 6 months, last 6 months, and they had the lump. So once it is following up, so they had the lump. Now we are observing and keeping track of the patient after each 1 month, that what is happening, what changes is happening in the genes, where okay, this may be quite advanced for you if you don't understand, but let me just explain in simple words, that when we see that how genes are expressing in normal tissue of the same person, then how the same genes, because genes count will be same, the same genes will be there in the person, so how the same genes are expressing in the lumps, and after 2 months how the genes are expressing, so how the expressions are changing. So this is how we are keeping track on the patients, and then we will be like we are studying with the scope that we will be finding that stage where the normal expression of the gene is changing into to translate to the cancer form. So that's how it basically goes. That's the overview, and there is a lot more inside. So how we have integrated is there is a bioinformatics thing, then there is a machine learning thing, and all things together. Yeah.
Okay.
Ma'am, we have some questions from YouTube Live as well.
Okay.
How has immunotherapy advanced as an effective and efficient treatment for cancer?
Immunotherapy. Okay. So, as I said that there could be immunity conditions as well for having cancer, right? So for that also, like we need to first detect. First, we need to obviously get the tumor samples, and we need to detect that what is the different is it the immunity factors that happens, which is the cause behind this cancer, and then accordingly we need to go for therapy, so every like advanced study of the cancer starts from the diagnostic testing. So for first procedure we need to go to diagnostic testing, and then we will understand that what is the particular factor behind this cancer, because once cancer happens, then we can go for testing, diagnostic testing, and then we can understand. But genetic testing is there where we can understand, read the genes expression and the mutation, we can understand. But till the cancer is not happening, there is no way we can understand that okay, this person is prone to cancer. So that's how once it has happened, then we can understand what factors are there, like is it immunity related, or related to what, due to the mutation or what exactly. Then we can go for further therapy accordingly, whatever is the requirement.
Okay, ma'am. Ma'am, there is one more question. Can
Mhm.
metastatic cancer cells become dormant?
Yeah, actually, metastatic. In that case, the cancer is like over all the body, right? Like different sections of the body, not specific to that area where the cancer is like supposed to be. So in that case, it is having the like it is prone to be dormant. Yeah, we can say that.
All right, ma'am. So the next question is, please wait a moment. Yeah. Does epigenetics play a role in cancer? That is the next question.
Yeah, epigenetics obviously play the role in cancer. In fact, we can say that that is a different part in a cancer study, because as long as we are studying the gene expression and then mutation, so in that case we are not giving focus on the epigenetic section, but everything starts from epigenetics level itself. So there is different stages how it starts, right? So epigenetics definitely play its major role in cancer. That is, we can say, the base of starting the cancer. It's just the study of the study level which is at the base level of where from where the cancer starts.
All right, ma'am. So, yeah, so mainly there is another question.
Yeah.
Mainly, we are all studying and working from our home. So,
Hmm.
is there any way we can, matlab, how we can pursue our research or work on something from our home related to cancer biology?
Okay. Cancer biology. Okay, so first, you guys need to understand or need to know that what skill sets that you already know, then what career option that you want to choose, that you need to be sure, so that accordingly you will be going to learn the skills, right? Whichever the career you want to choose. And then next, whatever once you have chosen, then you know that what these particular career require, that what skill sets it require. Then you can try to connect with us, even because we also provide like research, cancer research projects, so you can be trained and you can do projects. And there are many other like bioinformatics related or cancer genomics related companies as well, like bio curation is there, and other companies are there who provide the like research projects option, then training options are there, so there you can join and then you can just do the projects. Yeah.
All right, ma'am. So, the next question is what are your views on the CAR T cell therapy for the cancer treatment?
Sorry, once again.
What are your views regarding the
Hmm.
CAR T cell therapy for the
Yeah.
cancer treatment? Yeah, that is the question.
Okay. Okay, so that is again the immunotherapy, right? That is used especially again the T cells which are there for like responsible for cancer. So I think that again varies from what type of cancer is there. So because you are learning, studying a specific type of cancer, so if it is required for your study, if your cancer samples are having like a requirement for to study the T cells, or you have the samples from T cells, then you can do the like CAR T cell analysis and therapy. So yeah, that actually depends on the type of cancer that is under study. Yeah, and that is also one of the like good therapeutic strategy. Yeah.
All right, ma'am. So, there are actually many questions also. Then,
Oh. Okay, so what you can do is you can just also share, because probably what is the time of 10:20. You can also share, like make a list of the questions and you can
mm-hmm.
share to us, so that we can like answer all the questions, because of course everyone need to be answered, right? They have queries every bit, so that you they can opt for best career for them, whichever they are interested in. So you can just list out the questions and share it to us, so that accordingly we can share you the answers. You can forward it to them, the participants. You have already the list of them, right?
Yes, we already have a list of them. So,
Mm-hmm.
and also you can also share your email ID. Can you
Yeah.
please share your email ID with the participants so that they can question you or ask
Yeah,
you
I
any your doubts?
think yeah, we can share that one. Yeah, here it is. I think it's visible.
Yes, ma'am, it is visible. So, those of you who have questioned and your question is not answered, please
Mhm.
please mail to ma'am. She can definitely help you out. And
Yeah.
also, yes, those questions
Or
that we listed.
yeah.
Yeah.
Or what we can do, you can just grab their questions from the chat. Okay.
Mm-hmm.
So
Mm-hmm.
because we can save the chat, right? So we can you
Yeah.
can grab their questions from them, because for one question they will probably think that it is harder to email, right? We know that how frequently we use email. So for one question, so you grab their this questions, and we can answer them. So you can make some Google form or portal where we can just display all the questions and the answers.
Okay. So can I send it here on the Google Meet chat box? Can I do that, or can I mail
I
you?
No, you can mail me the list of questions.
Mm-hmm.
So then, once I share you, then you can share to them.
Definitely, ma'am. We can do
Yeah,
that. Yeah.
I think that will be better because it will take longer, right? How many
Yeah.
questions are there if they are answerable? So if are like few, but
There
I
are
think if you're sharing a
actually
lot are there.
Yeah, there are actually 12 to 13 questions.
Okay, okay,
So,
but
yeah,
uh, if I get to collectively at one place, then I can explain it better. I think like yeah.
okay. So, can we send it here or in the chat box so you can see that? And
Okay, right now. Okay, okay. Let me just hope everyone have noted the email IDs because I have just stopped sharing. Or those who are left, you can share them the email IDs because I have stopped sharing.
okay,
So I need to ask to Sir that if he is there for the session. Otherwise, I can answer few questions. So where they
so,
are asking the questions, like on YouTube.
yes, mainly from YouTube and also from our
Mhm.
students, our college students also.
Okay, okay,
Yes, ma'am. So we are sending the questions here.
okay.
Please wait for a moment. Okay, ma'am. The questions are given. There were some redundant questions. You have also
Yeah, right.
answered
Seven
them.
are
Yes.
there already.
There was one question: how can students take part in public awareness for early detection of cancer? That is the
Yeah,
question.
right. So there are various, like they can join the teams. Like there are doctors also organizing or social, health fair camps, healthcare camps, and then there are NGOs also organizing healthcare camps. So they organize for early detection of cancer or how to prevent cancer, something like that. So yeah, they can join that.
Okay, so how can I do research on cancer from our home? You have already answered that. Um, huh, okay, so I think you can answer this one as well because that is relatable to you. Rudy. How difficult it is to learn about the world of cancer coming from a different background altogether?
Okay, so basically, we should not think that how difficult it is. You can say it more beautiful way that how challenging it is, because every nothing is difficult. If you are willing to learn, you can learn. It's just that you should be interested in that. So and yeah, that's it what I can say, because I am also from computer science background and I am doing totally like cancer genomics. So that is also different for me, but I don't know from which background you are coming from. But overall we can conclude that if you are willing to learn, you can do anything.
Okay,
Okay.
ma'am.
Devan,
Yes, sir.
I think we should proceed with our next program. I think the questions, the students who asked the questions wanted answers from Madam can directly contact her over email. Is that possible? She has already provided us the email.
Ma'am, is
Yes,
that
they can
Yes.
connect with me on
So,
email.
so they can directly approach her, and I don't think she will be very much rude at all. The way she presented, so interesting way. I think the interested students might directly approach her for
Yeah,
any
sure.
questions.
I will. They are most welcome. They can
Uh,
email me anytime for any query.
yes, yes, ma'am. And thank
Thank you,
you
sir.
very much. That was a very fascinating session.
My pleasure, sir. Thank you so much for giving me
Yes.
opportunity.
Devan, I think we should proceed with the next speaker. I think,
all right, sir?
yes.
So thank you so much, ma'am, once again for sharing your expertise among us uh,
Most
and
welcome.
addressing the question from the listeners. Yeah, that was very much. We are very much thankful for that. So this brings the end of the first webinar of our morning session. Now we would like to introduce Dr. Inamul Hasan Madar as our next guest speaker. Dr. Inamul Hasan Madar is an adjunct faculty in Savita Medical College and Savita Dental College, Kuthambakam, Tamil Nadu. He is also a visiting scientist specialized in cancer proteogenomics at the Center of Proteogenomics Research in Korea University, South Korea, and Department of Biomedical Sciences and Environmental Biology at Kaohsiung Medical University, KMU, Taiwan. He was a vice president and core committee member of ISCB Regional Student Group, RSG India. Dr. Inam has graduated from the University of Madras in biochemistry with applied biotechnology, followed by a master's degree in bioinformatics from Bharatidasan University, Tamil Nadu. His MPhil is in biotechnology and PhD is in biochemistry with a specialization in clinical oncology research. His areas of specialization are precision medicine in clinical cancer research involving genomics, proteomics, proteogenomics, pharmacogenomics, computer-aided drug designing, in silico toxicology, and metagenomics in gut microbiota probiotics. Dr. Inam has authored more than 35 publications in high-impact scientific journals, including OncoTarget, Cancer Research, Analytical Chemistry, Nature, and Cell, and has worked on nearly 15 books in collaboration with national and international reputed publishers. Additionally, he has patented 6 of his major projects registered under Intellectual Property India. Other achievements include proteomics software development, where Dr. Inam has developed various proteomics-based softwares, namely multiplexed post-experimental monoisotopic mass refinement, to increase the sensitivity and accuracy in peptide identification from tandem mass spectra of co-fragmentations. He has also developed another software, Phospho-UMC Filter, a simplified method of localizing phosphorylation site using unique mass classes information to differentiate phosphopeptides with different phosphorylation sites, and multi-stage database search method, which is a data mining methodology developed for comprehensive proteomics data analysis to complement whole transcriptome sequencing data. These methodologies are now being used in Nature and Cell publications. He is also a principal investigator for 2 international projects from the Saudi Arabia government and has run various projects in collaboration with universities in Korea, Saudi, Nigeria, Taiwan, UK, and USA with Professor Nelson, Professor Gideon, Professor Hans, and Professor Iftikhar Aslam Taiwan. So, sorry, sir, for the introduction. Continuing, Dr. Inam is founder director of Hospital and Research Company. Currently, he is CEO and director of Sana Hospital and Healthcare in Tamil Nadu. Dr. Inam has recently initiated his two startups, namely Edomics, which is a bioinformatics-based company for research project trainings, contract-based research organization, and education and abroad consultancy for the students, researchers, and professors, and other one is Cancer Genetic Diagnosis and Treatment Company, that is Theragnosis Lab, which helps to get personalized treatment for cancer patients utilizing high throughput genetic testing and targeted therapy approach. We at SABBIT are really privileged to have Dr. Inam here with us. Over to you, sir.
Yeah, thank you, Ridhiman. So, can you able to hear me up?
Yes, sir, we can hear you.
Okay, great. So, Ghazala, you are also there now? Okay. So, let me share the screen. Just check it out if it's worked fine. Mhm. So, is it fine? Is it visible?
Yes, sir. Perfectly, sir.
Okay, great. So, let's go ahead then. So, hello everyone. I think thank you, Ridhiman, for giving a very good introduction. Actually, I think like there are a few other faculty members also there in that, right? So let's have a talk. So I just wanted to make sure before I start the presentation that let's not be like one-sided, just like a presentation, like where like usual, where we just sit in home and then like just listen to some audio. Okay, so let's have some discussion on like the topics that we discuss. Okay, so like I mean like I don't want anybody to be like very silent or something like that. Okay, so I think in your classes also there are some people who are very silent, not asking many questions or something, right? Like Sumitro, Rupsha, and other people. So you guys also have to ask some questions, and when we are discussed, and you can feel free to stop. You don't need to ask questions only at the end of the presentation, whichever like slides and information that you find interesting. So just go ahead and you can stop me and ask me. So like we'll be having the interactive sessions, not just like presentation. Okay. So do you agree with that? I cannot able to hear anyone.
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
There are 56. I can hear only 4. Yes, sir.
Very nice.
Yes, sir.
Okay, great. Let's go ahead with that. Like, let's make interactive sessions so that like you can have, even if you want, you can unmute your mics so that like whenever you feel like we'll have a discussion on it. Okay. So that like you can able to learn from things from there, not just on the presentation or something. Okay. So my aim is not to show the slides. Okay. Not to tell about what is there in the slides. I just wanted to, that I can do it by myself. Like if you guys are there, then I just wanted you guys to have involvement when we are talking about some topics. Okay. So we'll start with some very basic one. Okay. So very basic. Like I think Ghazala Sultan has really explained well on few aspects of like carry in cancer, and then like what are the other things that we can able to do in cancer, and there are a few other things like what we do and what other team people do in the group, and then the latest advancement and other things as well. So I'll be concentrating on my core area of research work, okay, which we work on, which is like a very advanced system of like cancer research, which is like on precision medicine. So which is my specialization in particularly with cancer proteogenomics, okay. So which we work on like in Center for Proteogenome Research in Korea University. So Korea University is in South Korea, not in North Korea. Okay, so I think everybody asks like, where is it? South Korea, North Korea. So we are from South Korea. Okay, where I did like most of my research work there. Okay, So is it everybody know where is South Korea? Anything about that?
Yes, sir.
Nobody knows. Who knows?
Yes.
Anything interesting from South Korea?
Sir,
Anybody
apart
knows?
from the Korean dramas and K-pop music,
Okay, that was always the first line I used to hear from everybody. Okay, that's good. So yeah, we have like different kind of things, like very good advancement in few things, like for example, Samsung mobiles, right? And Samsung medicines also there. Samsung hospital we have, and then we have LG, which produce like a lot of different kind of like displays. Okay, and then we have a best university in the world, like Seoul National University, Korea University, and Yonsei University, which comes around like almost less than 50 ranking in the world. Okay, so and then the Korean University, which is like I mean like sister university of Harvard and MIT. Okay, so where we work together on different projects, and we also play games and everything, as we are the sister groups. Okay, so where even if you go through the Korean University, you can able to see a lot of buildings that are like very similar to Harvard and Oxford. Okay, so that's very interesting. Like if you are interested to discuss about more about the university and other things, I'll be really happy to do that. Okay, so let's move
Sir,
on to yes, please go ahead.
yeah, I heard a fact about South Korea that there is a very popular destination. It's a very popular destination for medical tourism. Can you please put some light
Yeah,
on that?
exactly. Yeah. We have very good medical tourism there. Okay, and where we have very advanced system of like medicine that we work on. Okay, so there are few medicines that we work on research basis that also we include in the treatment as well, but mostly it's but we have like for example, this is a kind of like precision medicine that we started. So we have in our own hospital, we have like Korean University Hospital, Korean University Medical Center, where we give a treatment based on like precision medicine, and we are the core people in the whole Korea that we started precision medicine using like proteogenomics technologies and other things with the collaboration from NIH USA. Under that, there is one institute called as like National Cancer Institute. Okay, so we have a collaboration directly there from where we get the fundings and other things as well, and we also implement those things in few different countries. So hence Korea is one of the like very good hotspot for the better treatments. But it could be better, like because if you are Indian, like we'll be always talking about like how much it costs, right? So in that case, like if you have a kind of international insurance or something, then things gonna be very very good. Otherwise, it's gonna be a little bit expensive than other places. But the treatment is really of very good quality. So you will not have any kind of like feel that okay, this kind of treatment is not there or this kind of equipment is not there. So we have most of all the things equipped there. Okay, which is almost leading when you compare the whole world, you can able to count our hospital into top 10 in the world where we are giving the treatments. Okay, but recently there are some people from Delhi and Mumbai and Bangalore. So they have started helping patients to move South Korea for the treatments there. So people usually get like medical tourism as you were talking about. So we give like different kind of treatment, not specifically for cancer. We also give like kidney transplantation, liver, and other things as well, which is usually like recently I mean like there are a few hospitals in India. I think most of the people move from north to south to get operated or something like in the recent time, I mean. Okay, okay, shall we get started with the presentation?
Yes, sir.
Okay,
sure
so
thing.
Sure.
Yes,
just
sir.
one query I have. I think like there are a few faculties as well, right, now in the presentation. I think one or two faculties like Nara, I mean Dr. Narayana and Mr. Narayana left already because he was busy.
Yes, sir. Faculty is also there.
Okay. Okay. So any other faculty is right now?
Sorry, sir.
Any other faculty member right now in the session?
Yes, biotechnology faculties are there, sir.
Ah, okay, great. Okay, that's good. Okay, so yeah, that's really nice. Okay. Okay, so let's get started with few things. Okay, um, I'll be having some queries as well during the presentation session. Okay, and anybody wishing to answer, they'll be happy. Otherwise, I can name some people who can answer as well. Okay. So, like first thing. So I think like as we are talking about whole day is about like cancer. So can anybody tell what actually is the cancer? In a general term, what does it mean even?
Sir, cancer. I'm sorry, sir. Can I speak, sir?
Yeah, please let me know your name, please, so that I can able to know who is talking.
Sir, actually, my name is Kaustav Datto. I'm from BT second year of our college,
Oh, that's
Bengal
great.
Institute.
That's very nice. Go ahead, please.
Sir, cancer is a group of cells which was
Mhm.
actually before a tumor which has undergone malignancy and is in the state of, it's in a state of highly increment or highly division which cannot be
Mhm.
stopped or which cannot be controlled, or if it needs to be controlled, it needs medical treatment.
Okay, that's great. Okay. So any other questions? What about Sudeep? You have any answer? Avjit, what about you?
Sir, basically it's a uncontrolled cell division that can
Mhm.
lead to some disease, or it can spread and evade other parts of the body.
Okay, that's really nice. So, do you know when is the Cancer Day, Nikita? What about you? Do you know any idea when is the Cancer Day in the year? When we celebrate, or when we there is something called as like Cancer Day in the whole year. Do you know, Nikita? Nikita is not there.
Yes, sir, I'm there. So
Okay, what?
it is celebrated on February 4th.
Okay, you're googling. That's good. You're very
No,
good in that.
sir.
Okay, okay. So, okay, that's nice. Okay, so yes, you're right. So that's what that we celebrate the World Cancer Day on like 4th of Feb. Okay, so let me come to the question that I was asking about. So, what is cancer? Okay, so in general term, in a colloquial term, not in the scientific term, if you wanted to understand what actually a cancer it is, cancer is like something which is uncontrollable. Okay, so for example, to better understand in a colloquial way, is that like someone in the classes, okay, so who will be keep talking too much, okay, so it has like talking cancer, or something like someone is eating like usually in our terms like something uncontrollable. It's not like into limited. Okay, everything is good when it is limited. Okay, and even the good thing is if it goes beyond, like if you talk about the general like if you talk about the cancer cells, okay, normal cells, okay, so there will be in the initial stage, so the division of the cells or the multiple of the cells, there should be a normal growth. If the growth is of little more, I'm talking about a colloquial one, not in the exactly molecular level or scientific level, just to understand, like to know what actually the cancer it is. Okay, it's something which is uncontrollable. Okay, so that is called cancer in a normal world. Okay, so we have different kind of things that we'll be discussing in the further slides. Okay, so anyway, any idea what causes cancer? Sayantika, any idea on that? I mean, what causes
Yes,
cancer?
yes, sir. It can be causes by any uh, mm, environmental effect or any other genetic uh, mm, abnormality. That is a P53 gene. We know that the controls the tumor. mm,
Tumor
the
suppressor gene,
conscious.
yeah.
Sorry.
That is P53 is tumor suppressor gene.
Yeah.
That is one of the important pathway that we can able to draw for the cancer. Okay,
Yeah.
so I'm just telling what causes exactly. Any idea?
Sir, can I speak?
Yeah, yeah, who's that?
Sir, Kaustav, sir.
Oh, yeah, yeah. Please go ahead.
Sir, actually, a cancer is caused when a cell loses its contact inhibition And
Contact inhibition.
yes, I mean, when it is surrounded by other cells and it loses its ability to remain intact in that size, and it spreads
No, it's
uncontrollably.
cannot able to tell it's intact in most some cases because it's not in always like that way. Okay, when you talk about the etiology or when you talk about like the actual molecular level of the cancer or cancer cells, okay, it's not intact in most of the cases. Okay, so when it compared to that of benign and malignant, okay, so then we have a different kind of scenarios in that. So it's depending on the case by case. But as we are from biotechnology, right? So I think everybody is from biotechnology. Am I right?
Sir,
Yes,
it
sir.
mostly happen because
Yes,
of
sir.
mutation, any kind of mutation.
Okay. So any other I mean, any other graduation? I mean, like I mean, some students are there who is other graduation from biotechnology here in this system.
Sir,
Uh-huh.
sir, the cancer can be caused by carcinogen, cancer causing agents. Like,
Okay.
there
So
can be
okay.
various
So
factors for that.
yeah, that is carcinogen, but that is from the external factor. What about in the case of like, okay, being be as a biotechnologist or molecular biologist, then answer about what causes cancer. I mean, we need to be like because what we are originally from biotechnology, right? So we need to tell that like from our perspective, what we have when we are studying about molecular biology or genomics and proteomics or other things, right? When what our professor was taught teaching in the classes, so we'll have a pretty idea what can be causing the diseases. Okay, so let me make things easier for you guys. Okay, so these are the few things. Okay, for example, a gene responsible for that one. Okay, so because we are from molecular biology or genetics or proteomics, the main thing which causes cancer can be genetical modification or other informations. Okay, so but many people in India, they think that they have a different kind of myth. Okay, where many people ask me this kind of questions. Do if I mean like if I use more kind of cosmetics for the girls, and then if you're in Korean, then like even for the male, for cosmetic, and then like if you use like food plates or plastic bottles or something that causes cancer, or mobile phones, that is like very commonly you can able to hear for almost from 99% of people that mobile phone causes cancer. Okay, that's what people usually ask me, but it's is a myth because there are already some people who did research on that. They haven't found any connection with that. It may be related to cosmetic, it may be related to the use of plastic, it may be use of mobile phones. Okay, but yeah, there are some there which is like kind of a diet, obesity, alcohol, smoking, or even like huge of amount of like UV radiation. Okay, it may be from the sun or it may be from the chemical hazards. Okay, so whenever something like you, because whenever you are attending some sessions, okay, like presentation on some particular topics, you need to have some point which you can able to take away home. Okay, so where you can able to have very concrete idea, like this all kind of myth, which doesn't cause cancer at all. Okay, because there are some research work which is already done, but there is no results out of that one which has any linkage in that. There is no like research identification for that. Okay, but yeah, for the smoking, alcohol, obesity, and diet, yes, there are some proven risks are there, and where you can able to find a lot of research works and research publications are there where we can able to tell that this causes cancer. I mean that one with the physical external efforts, as well as like we have changes in the biochemical pathways and biochemical things and other things as well. Okay, so make sure this slide is completely on your mind. Okay, where if somebody is telling you, oh, I hear that one, like this one causes cancer, that one causes cancer, there is no proof for that. But this all the myths are mostly from like Asian countries, like where people are talking about this one, but it's not at all proven. It's just a myth. Okay, or people just tell like like some people who are not who are too addicted to mobile phones or something, right? So people just say, oh, if you use the mobile phone then you will be getting cancer. So it's kind of like making someone fear to not to do something else, to tell something other things. Okay, but scientifically we need to have some like clear idea about what causes the things. Okay, Okay, so when we talk about like the estimated new cases in the case of like 2021, okay, the most like time, the most major times in the case of like female. So I think you guys can able to see um, my this one. So in the case of like male, the predominantly will be prostate cancer. Okay, where in the case of like female, so mostly they are suffering from breast cancer. Okay, which Ghazala is expertise and I mean her own core area of research work. Okay, but when you see the mortality where the people like causes of death due to cancer, okay, it is because of like lung and bronchus. Okay, so in this case, like we already helping people to treat like prostate cancer 100%, no doubt. Okay, so we have some report that we couldn't. We were asking permissions from the patient now, so that like we can able to show some students like how the prostate cancer has been getting cured right now. Okay, so the patient was like started suffering from prostate for almost like 7 months before. Okay, and then like you can able to see like there is really lots of I mean cancer cells in his body. Okay, so I don't have any permission as of now to get. I don't have concern from the patient, and he told he's okay, fine, but I just need to get the signature from him to show the result. Okay, so if later on, if the next presentation, if you get the chance, I'll be really happy to show you guys. Okay, so as this presentation slide where I will just wanted to emphasize about like how a male and the female are differentiated when is the incidence of the cancer. So where in the case of like female, like predominantly, you can able to see almost like 30% out of 100, like 30% of the female is going to be suffering from breast cancer. Okay, so in India, and then like in the case of like male, there is 26%, and then it's started to work very aggressively. Okay, but when you talk about the mortality, it is not the breast cancer or the prostate. Okay, so mortality is most of the times like it's due to lung and bronchus. Okay, and it's equal to both. It's like 22% in the both of male and female. Okay, so that's also we working on and like lung and bronchus cancer, so like we could able to save patient as much as possible. And this all the thing is not just like okay, always consider that one. It's not like completely we talk about the cancer, death, and this all things. This is not only from the biotech, I mean like medical background. There are many people who work on bench level, like as like you guys, okay, like biotechnology people and molecular biology expertise people who work on bench level on the cancer cells or the like specifically on the breast cancer, prostate cancer, other a different kind of cancer, and they produce different kind of results. Okay, so which helps people to get better understand the cancer and then target the cancer cells and treat them well. Okay, so I mean like biotechnology people or molecular biologists and other people, they stay as a backbone on all this kind of conditions, because when people usually see like they think that okay, there's something like normal cancer, like some other doctors will be doing it, but the people who are working behind are molecular biologists, like microbiologists, okay, or chemists even, okay, even from different backgrounds, they work on that one together to get a very good result out of it. But where the biotechnology is like candidates plays a very pivotal role in helping and identifying or like researching on these aspects. Okay, let's talk about what are the common type of cancer like usually that we get. As I told about like lung, liver, and colorectal, breast, stomach, and cervical, which is a kind of like whole in Asia. Okay, when you talk about the whole in Asia, the still like, but in the case of men, it's like from lung cancer. But if you talk about specifically for India and other country, like mostly predominantly is like prostate cancer. Okay, but in the case of like whole Asia, so which is like lung cancer is a predominantly occurring one. Okay, so what are the leading like estimated in the throughout the world? It's a different one. Okay, where we have like prostate cancer and breast cancer with a new estimated for the cases, and then mortality as a lung and bronchus as well. So we can talk about like India, Asia, and then for overall population for the new estimated, and then like this one. So now, as I was talking about, like what you guys think, which causes cancer, as being from biotechnology background or molecular biology or genetics, so mutation plays a very, very important role. Okay, which when it started to express, it started to shows and started to make function of that. So you can able to feel there is some, what do you call, initial or progression of cancer. Okay, so just let me come to one more point. There was someone who have asked, like, what was the question in that? Okay. What was that? What was the seventh question, right?
Sir, how long does it take a normal cell? Yes, in the chat box,
Yeah.
right?
So how long it takes a normal cell to acquire the appropriate type and then number of mutation to become. Okay. So first thing, let me make it clear, because the question, who was the question? I mean, who was this who asked this question? Can you name them?
Sir, actually, there were too many questions, so we couldn't keep track of who asked the question. So
Okay, no, I'm just asking because everyone is here now, right? How long it takes a normal cell to acquire the appropriate
okay.
type of a number of who asked this question, so I can able to like explain them.
So I asked the question.
Name, please.
Sir Shudeep.
Okay, yeah, actually, yeah. So actually, mutation exists when you birth. It doesn't like. I mean, do you think that it's possible? Like, if you're talking about like genome editing and other things, which has not come into reality and picture till now. Okay, so you cannot able to mutate a gene. Okay, so it exists from the birth, but it's depending on the timeline, different depending on the expression and everything. Because some people even ask me, I mean, if you use in the chemical industry, for example, or in the radiation or something, there is a very rare condition. For example, if you need to have a very strong ray, for example, X-ray or something. Okay, in normally you cannot able to get. Okay, for example, till 20 years I was fine. Okay, then suddenly, like after 20 years, I got one gene mutated. So it never happened. It's already exist there from the birth. But the thing changes is that that particular mutation or that particular protein started to function. Okay.
Okay.
It depending on the time when it started to function, not depending on like it started to mutate at the time. Okay.
Sir.
And in the extreme condition, okay. Yeah, someone was asking something. Please go ahead.
Yes, sir. Actually, I'm asking just a small question. That actually you told that type mutation which you are undergoing is present from birth. That means if someone is going to have cancer in his later part of life, this can be misunderstood from his birth.
Exactly. But one thing, okay. So what you can able to give the guarantee that particular mutation is going to express in his lifetime or not? There is a mutation exist, okay, which causes is in a cancer gene, okay. I have seen, for example, if you talk about like Mendelian, right? Mendelian theory, right? Like where you have recessive characters and the dominant character, right? And then some are like carrier, right? So some people have the mutation as a carrier from the parents. If you talk about, there is some case studies, okay, where grandfather parents have expired, okay, due to some, like for example, like leukemia or some other diseases, okay, some other cancer, okay, and then where the parents are fine, okay, for the mother both are fine, and then the children's also fine, but the grandchildren's have those mutations and then they are suffering from leukemia, for example. Okay, so that means that these people are carriers, but they didn't express at all in the whole lifetime. You understand?
Yes, sir.
Yeah, great.
So,
Please
Haley
go
here.
ahead. Yeah,
So
please.
is this applicable for the external factors causing cancer as well? Like,
So
if I'm
which
prone to the mutation already, the external factor affecting my body can also like trigger the mutated condition.
so you mean to say that external factors can able to mutate your cells?
Yes, sir.
So for that one, you need to have a very hazardous material. Okay.
Okay.
So that, for example, a huge amount of UV rays may change. But one thing, let me come to the direct question of it. So your question is, can the external effort or external things or the environmental factors has an application. Okay, it can causes, but
Yeah.
that is not the primary thing. And that is like not happen in 100 cases. If you talk about mutation is in 90%, okay, 92%, 95%.
Okay.
Okay, the one which you're talking about, it may or may not be. Okay, but
Okay,
in
sir.
this case of mutation, it is for sure it is there, but it express or not, it's depending on the person's like homeostasis or like or the physical conditions and other things. You understand?
Yes,
But
sir.
external factor. Okay, let me talk about one condition where, for example, someone is suffering from who is working in a very like a chemical factory. Okay, obviously he will have a cancer. Okay, but the thing is, he will have a cancer of multiple myeloma, which is of a type of skin cancer.
Okay,
Understand?
sir. Yeah,
So
I
on the
get it.
skin cells, we get a contract. Okay, and because of he's working in a chemical factory, we cannot tell that he got the liver cancer. You understand? It's both are different
Yes,
because
sir.
even if you're using a strong chemical in the laboratory, and your skin started to have some effect on that. Okay, the chemicals.
Okay, sir.
Even in the cosmetic, if you talk about like high or very low pH or something, then you have an effect on your skins. Even in the cosmetic products or some other thing. Okay, because many people are talking about like even in cosmetic it does, but it's no at all because nobody make like that kind of things, which can affect too high. Okay, it's a crime to do. Okay,
Yes, sir.
yeah. Okay. Any other questions? Feel free to ask. Okay, it's
Sir,
just not from. Please go ahead.
Sir, mainly during gel electrophoresis, we use a chemical etherium bromide, which is said to have carcinogenic activities, or it is
Yeah, that is like
treated as a
laboratory.
carcinogen. Yeah,
Yeah,
yeah.
that is
So,
carcinogen. That is laboratory level. It can
mhm,
causes. Okay, but you have
mhm,
limitations of like there are some internal as well. There are some like external. Okay, for example, like which is skin cancer.
mhm,
Okay, it may causes because of the chemical. That's what the explanation I was giving. Okay,
yeah.
ETB or other things that we are talking about.
Mhm,
Okay, it's it causes as well. That's kind of a skin that has a limited one. Okay, because the reason why, again, let's go to the molecular level. So it changed the structure of the cell or ruptured some cells. Okay, it is not for the whole body.
yes, sir.
So, what are
Yeah.
the chances of one getting cancer via natural causes than like external factors? Like, what are the
So as
ratios?
what we can able to say that external factor we can completely like make it to like 1 to 5%, for example,
Okay.
because we are being into biotechnology. Okay, we will be talking about with the evidence of what our own area of study, based on what we have studied, what our professor has been taught. So, like in the classes, right? You're in the second year now. So your professor, who are talking about like different kind of mutations which cause diseases and other things as well. Okay. So because other things that we can able to discuss, okay, externally, we don't have some sufficient proof for that. You understand?
Yes, sir.
But the things we're talking about the mutation, we have the information about the mutation in there, and then what is the pathogenic level of that, and then it also has a specific drug target for that particular mutation, which can cause us cancer, and we have a metabolic pathway as well, where it can be defined that because of this one, this expression level or inhibition or suppression happens. Everything we need to talk about the whole thing. Okay, because we are into scientific environment, We are not people like who are into different field of study, right? We are not into business. We can tell that, okay, oh, don't use the cup of the coffee, or which is the plastic, you will cause the cancer. But we need to have some evidence proved. I'm talking about the theoretical, which is proven, right?
Yeah.
External is there, but what is the proven record of that? People did research work on that one. Okay.
Okay, sir.
There are few research work which has did, they have already produced a negative result. But in, like, for example, they take 10 to 50 samples and they worked on it. So they haven't found anything. And one they found, maybe they also think that there will be a technical error. Maybe if you are going to the way, if you support your question, for example, okay, so maybe 1,000 to 2,000 of a lakh samples we need to take, maybe one chances. So we will be concentrating on, there are something, if you talk about 10 patients who are suffering from cancer, all the 10 has a common thing which is called specific mutations, which causes specific kind of cancer. So which one you will be concentrating on? Right?
The mutation, like mutation.
Yeah, exactly. So because we can able to target that one, okay, we can able to design a drug, okay, we can able to work on the pathogenicity of that one, we can able to draw a complete metabolic pathway and other things, okay, very clearly, so that we can able to have a clear understanding how the initiation or the progression of the cancer happens. Okay.
Yes, sir.
We have a complete record of each and every mutations that we work on. And there are like thousands of research work which supports each mutations and other things. Okay. I mean, I'm talking about, like, there are other things out there which you're talking about, external things. Okay. So, which is kind of like, I cannot able to give you a strong word on that one, because there are not much like research work has been proven. But I'm very sure that there are some research work has been done based on those kind of hypotheses. And it has already proven it's ended in the negative things. Maybe, I'm not sure. Okay. But the mutation part, being from the biotechnology or molecular biology staff, we are 100% sure about this one, because we have seen those kind of things. Okay. Like, for example, in the case of like breast cancer, BRCA is there, BRCA as a drug target. Okay. So now we can able to tell that, okay, no, you have drink tea or coffee in the plastic mug, right? So that's not like a kind of an educated person or scientist can talk about. So it's about like exactly what I have a proven data, right? So yes, when you talk about something, most important is like because the difference between educated and an educated person, or a normal studies, or a scientist, the scientist, or you guys also can be a budding scientist, okay? So in that case, you will be talking about the things which is already have a reference of that. A reference in the same sense of like research publications or the patent, not from some blogs or messages from the WhatsApp, okay? Which has no reference, right?
Yes, sir.
Yeah, exactly. So that's how it works, because we need to be very clear on our idea. So I'm from biotechnology and biological background, so I know the main thing, which majorly, predominantly, let's not make it 100%, okay? 95% is because of mutation, because I'm coming from the background, okay? Right?
Of course, sir.
Yeah, great.
Thank you, sir.
So, yeah, always welcome. I'm
So
really
another
happy to
question, sir. Sir, Prerona here.
Yeah. Oh, Prerona. Yeah.
Sir,
I was waiting
actually,
for
you
your
talked about.
questions.
Sir, you talked about the plastic bottle thing. So another thing, nowadays we all tend to heat our food in microwave ovens. Sir, how much do you think these microwaves can be carcinogenic for our body?
It's not about thinking. Okay. It's not. It doesn't matter what I think. Do we have like proven data? Okay, research publications on that one. That's the question. Okay, let me tell you one thing. Let me tell you, because I can be for an hour with you right now. Okay, or some 30 minutes or even 2 hours. So after this, maybe we may not be able to contact. Okay, but you can able to ask yourself what amount of the evidence that I have for something I heard. Okay, so when normal people are talking about something, okay, like for example microwave or the plastic which we use in the microwave or something, okay, it may causes cancer or something. Okay, so first, okay, if you talk about like scientifically in the microwave, for example, it just make heat up, right?
Yes, sir.
Am I right?
Yes,
So
sir.
what is the ray which passes there? Is it like microwave passes X-ray or hazardous rays? No, let's talk about scientifically. Do you think that like something within a fraction of 30 seconds, a very harmful will be there? And you know, like if according to the theory that we go with your point, okay, so it's like whoever have the microwave will be dying. Everyone. Okay, right?
Yes, sir.
And everyone will be suffering in the family, like
Yes, sir.
with the cancer and dying. And in the whole home, only the microwave will be left. Right?
Yes, sir.
Now I'm talking about the practical aspect,
Yes, sir. Yes, sir. Understood.
right? Because this all kind of myth which has no proven, but I can able to make very sure, okay, being in our own study, right? So we are from our own departmental study, what we are studying right now in our syllabus and everything. According to that one, okay, mutations plays like very pivotal role. Okay, it's a proven completely, and we do research for years and decades. Yes, please go ahead. Yeah, someone was asking some questions. Hello? Hello?
Sir, you're audible,
Hello?
but
Yes, sir, you're audible.
Yeah. So I thought someone was asking questions. Okay, let's move on to the next slides. Okay. So how gene mutation occurs? Okay. For example, if there are two people, okay, so you can able to see here. Okay, is it visible? No. Right. Okay, let me make it this way. So this is a chromosome, right? Any one chromosome you can take. If you talk about the gene, then you can able to see there is one. This is a normal one, blue one. Okay, let me make it like normal. Okay. Okay, and this guy is suffering from cancer, for example. Okay, so the normal is like C, but this guy has the mutation as T from here. Okay, so which is a mutation occurred. Okay, if the mutation happens, a single base is altered. So this is kind of an DNA level. Okay, there is some 80. I mean, usually in the nucleotide 80GC 4, right? But I just wanted to make a differences in this. Okay. So here, okay, there is a, just to make you guys understand, okay. So this is kind of, I think everybody knows what is codon. Anybody have doubt in that? What is
yes,
that?
sir. Yes, sir, we know.
Okay, Ridhiman, can you explain that?
Sir, it is mainly a triplet of nucleotides, which mainly signifies a protein or an amino acid, which it codes. Sorry, it is not. It is not DNA. It is not. It is an RNA, mRNA actually. So during translation, these codon sequences are translated to amino acids, which eventually forms the protein.
Yeah.
Yes, so that's it. I
Okay.
guess.
Understand. Any other? Rupsha, you have any answer for that? Amartya, any answer? Is this guys are not there? So Ridhiman, can you able to hear me?
Yes. Yes, sir.
Rupsha, Amrita is not there? Okay. Perona, Sumitro, what happened? Even you guys are silent in the class, and here also you guys are silent. Okay. Okay, so I'll just move ahead. Okay, so the thing is, the three nucleotide from messenger RNA, okay, mRNA, can be a codon, which has the capacity, okay, to give rise to amino acid. Okay, so for example, it has a meaning. Okay, it's a genetic language, okay, which is a language of life, which everyone has it in within them. Okay, so if you talk about this particular sentence as the man saw the dot hit, okay, hit the can, and it is, so it got differences. Okay, but if you talk about only here, okay, it is dot, right? But actually it was dog which got mutated, right? So the meaning of the whole proteins differs. It was from the dog to dot. Okay, so when the meaning of the sentence is completely different, as the man saw the dog, okay, but here as the man saw a dot, both are very different meaning. So that's how the things difference is there. Okay, so it's normal, it's a very general term to make some people to understand how genetic information can be, how much it can be, meaning can be changes. Because when you talk about ATGC or TGC, not much people understand. Okay, but when you talk about the genetical language in our languages, so that this is normal thing that we talk about. A man saw a dog and hit that with something or something. Okay, but it's a dot here which is changed there. So you can able to see that just because of one mutation, there is the whole things has been changed from that particular gene or protein. Okay, so this is like a normal information. But if you talk about the how the mutation happens from the normal cell, okay, I mean from the normal cell, then if there is a mutation, it can be hereditary or radiation or chemicals or spontaneous errors during the DNA duplication when during the development. Okay, so mostly is because of the hereditary at the first. And second thing, which is already there, sometime what happened, we have two kind of mutations. Here you can able to, because we have added here as a radiation as well, because this one is when we use the experimental for the X-ray, okay, or infrared for the higher one, or ETBR, so which is a kind of a carcinogen which causes cancer. That's why we made it in the second one. Spontaneous error during the DNA duplication, which is kind of like during the metabolism or development of the fetus. Okay, and then those mutations, one is there. Okay, when it start to develop, it become two, and then it becomes 3 and 4, and then it goes on develop uncontrollable. Okay, so in that case, what happened is that, like, if there is a mutation and then there is a proliferation, mutation happen, a growth where the tumor formation is there, and then in that tumor, when it's high, it become like very malignant tumor. Okay, and it went to the bloodstream, it start to have energy to make a very good metastasis tumor. Okay, so that's why like angiogenesis is one of the important thing, what it plays important role in development of the cancer. Okay, and the most important thing is like how we can able to help people getting treatment and then get treated. And what is the most important thing? The most important aspect is that, like, okay, most important aspect of the here is, okay, so I'll ask you the question in a different format. Okay, okay, what will be the best way if someone got the cancer? Okay, according to this figure, what do you think that a best way to treat the patient and get cured?
Sir, in the early stage, the
Yeah,
early
so that
stage of
is
detection. Yes.
very important. Okay, because yearly detection of the cancer is very a best thing, and then we can able to cure very well. in a very short span of time. Okay, if you start to diagnose late, it gets extremely worst, and then it is difficult, but it's not like you cannot tell that okay, it's not at all possible. The first thing for today's session, you keep one thing in mind: cancer is something which is completely curable. We are not living in almost 30 to 40 years before, of 3 to 4 years, decades before. We have a very good advancement where we can able to cure cancer completely, zero. Okay, because that's what we are concentrating on, that's what we are working on right now, and we are seeing the patient every day getting cured. Okay, where some people, like some doctors, they have told that okay, this guy is going to live only for 15 days, 20 days, and afterward he's no more. We started to work on the patients, okay, and then we could able to see very good results. And like for example, like there is one patient just recently who is undergoing the treatment with us. So he came to us almost like 5 to 6 months before, where he was completely bedridden. Okay, and he's bedridden for about 4 months, and then he couldn't able to even get up or do anything, even urination, everything, fecal and everything, food, everything in the bed. Okay, and he was like almost every week start to reduce the weight. Okay, but the good thing is that he contacted, and then we started to diagnose, and then we did work on his reports and everything. Okay, but he was into early stage even still at that time. Okay, that time we started treatment, and just okay, he was, as I told, he was completely bedridden. He need even 2 to 3 people to make him to sit a bit, not completely sit like this X and Y axis. Okay, even for a bed it was so difficult. Okay, we started almost like 222 days now medicine. Okay, just last week he came by himself. Okay, and he was very happy with the treatments and everything. Okay, so this all the time things, kind of things are done not by the medical people, not by physicians. Okay, people from biotechnology, molecular biology, another pharmacology, right? So all these people are the backbone where we understand like how early diagnosis work, how the mutation can happen, and what are the different kind of initiation and progression, how we can able to stop. Okay, so all these things, we stand as a background. Okay, so the reason for telling that one is we have, as Ms. Ghazala was talking about, like career and other things. So you guys have very good scope. Okay, so where you can able to move forward and help a practical aspect of like what actually is really in need by the people. Understand? So do you guys have masters and other things in your college and in your institutes?
No, sir. We have BTech in biotechnology from our institute.
Okay, BTech in biotechnology. Okay, great. Okay, that's a 4 year course, right?
Yes, sir.
Okay, that's great. So yeah, so the most important thing as of for this one is like, I mean, we need, it's always better to go and diagnose yourself. Okay, so that's why in South Korea and other country we have yearly once mandatory health checkup. Okay, so that's a good thing that we could able to adopt those habits, so that we'll be having idea what's going on with and changes in the body. Okay, based on that one we can able to correct ourselves. Okay, and then if we can able to detect the biomarkers, okay, and then using the genetic studies, easily we can able to help to get the treatments. Okay, so the characterization of the cancer cell, this is one of the most famous like figure. Okay, this is like of very in detail. I mean, like how a cancer initiation and then how the progression, how we can able to inhibit. So this is kind of like a logo for the cancer research. Okay, which is like a very landmark paper which is published in Cancer Cell. Okay, almost a decade ago. Okay, how proliferation with the signal pathways, the cancer can be developed, and how we can able to uh, suppress the growth of the cell, and how it also activate or metastasis. Okay, and how it can be like how angiogenesis plays a very important role. Okay, if anybody is interested to know much more detail, I'll be really happy to share the paper link. You can go through. It's a kind of like very basic and bible of the cancer. Okay, how and what happens with the cancer cells, how inside play the roles and everything. Okay, so I'll share because it's too much detail on this one if you talk about. So let's move on the normal things. Okay, as someone was talking about ETBR, right? And then which is carcinogen and everything. We do have in the molecular level called as like proto oncogene, okay, tumor suppressor gene. So someone was talking about p53, okay, which comes under tumor suppressor gene, where if the normal proteins are there, which develop from the genes, okay, so normal cell division will happen. But in the case of if there is some defective in the protein, as we talked about like earlier, instead of dog, there is a dot. Okay, then the protein is defective. Okay, then you can able to see the uncontrollable cell divisions there. Okay, so even in the case of like proto-oncogenes, where the point mutation and amplification or the translocation, okay, so this also has an important role as well. Okay, where we can able to see the hyper growth or even in the uncontrollable growth as well. Okay, so let's talk about. Okay, this is all the general topic. When we talk about there is someone question what you think that we can able to do some research work by sitting in home because of the lockdown and no classes or something, right? Or can we able to do some training if you're interested or something? There are some topics are there, like clinical genomics that we are expertise in, and we are working on functional genomics, and then pharmacogenomics, pathogenomics, and translational genomics. This all the things that you can able to work on. So this all completely are integrated, and then it can help directly a cancer patient to have a better treatment. Okay. And then this is my main core area of research work, which is starting from the genomics, which is all genetic material of an organism, which is called genome, and then a full range of RNA molecule, which is tRNA, mRNA, and other RNAs, okay, together in organism, which is transcriptome, and then proteome, the entire set, which a protein, protein, which expressed by a genome, okay, which is called proteome. And then we have a phosphoproteome, where we have phosphosite localization or post-translational modification from the phosphosites, okay, in the peptides and proteins, okay, from the proteome. And then we have a phenome, which is phenotypic characters of the cells, okay. And then here we call as a proteogenomics, which is one of the highly advanced system of research work in cancer, okay. So now even NIH and NCI, where we have collaborated with, we concentrate completely on proteogenome research. Okay, and then we also work on how those particular functions, okay, and the gene product of those. Okay, so here when you talk about proteogenomics, we have genome, which is whole genome sequencing or whole exome sequencing. When we talk about transcriptome, we have RNA sequencing and exome sequencing as well there. If we talk about targeted codon sequences, and then this is what we work on here for the long time, proteomics, where liquid chromatography and mass spectrometry based proteomics, GCMS, TOF, and everything. So we develop and we work on all this aspect of research work with the cancer samples. Okay, and then if you talk about translational genomics in specific, okay, we usually use clinical biopsy, okay, liquid biopsy samples like blood, okay, for example, and we also help in identifying the therapeutic targets, stratification, okay, and then early detection as well with the liquid biopsy samples, and then we can able to get the real monitoring for the therapy, and then we can improve the drug delivery as well, okay, and then we can able to help so that metastasis could not be a relapse for the patient. So for this example, so we give the treatment for the patient for almost 11 years before, so who are suffering from gastric cancer, throat to gastric cancer, okay, the whole. Okay, we started to give, we did work on liquid biopsy samples, and we started to give a target treatment, and it's almost 11 years. So and we keep a record of her. She's doing really fine. She's almost now 77 years old. Okay, so like almost 11 years before she got. So I mean, she was suffering from cancer. We did our own technology, and it was really helpful. Okay, and now when you talk about like genomics, so what happened? We extract the DNA, okay, fragment it, and then we use different libraries using PCR, and then this is the Illumina machine that we use to sequence the whole genome. Okay, so and then we have like we add the primers, and then we develop a library of the whole genome, and we sequence it. And then after we sequence, we have the like nucleotide, right? Then if there is some mutation, we identify and get the other result. Okay, so this is all like bioinformatics aspect of that. When you talk about the example preparation for the proteomics, which is like LCMS, so we use like separation of the protein. First we take the protein and then digest that one using triptic peptide with the trypsin. Okay, where in the case of like separation based on hydrophobicity, which is HPLC, we use high performance liquid chromatography, and then we use ionization method. Okay, which got the Nobel Prize earlier by Dr. Fenn. Okay, then we like count on like parent ion and then like fragment ion, and then we use bioinformatics method to quantification of the protein and then identification of the protein using the databases. Okay, and we also work on like PTMs, okay, which is post-translational modification. So you can able to see if you talk about only this one with ATP and ADP.
Yeah.
Who is that?
This is Shantika.
Okay, yes, please. What is ADP and ATP?
ATP is the active form of energy that can be stored in the that can be actually used in our body. So
okay, from ATP to ADP, what is the things it is lost?
Phosphorus.
Yeah, exactly. Okay, so that is usually we call as like when in the protein sequence. Okay, this is the serine side. Okay, if the normal serine. Okay, with the side chains, which is like open for the phosphate group to be added. When ATP gets to add up, okay, and the protein kinase happens, where phosphorylation site is added. Okay, which is kind of like switch on for making a function, because in biological, in some part of the phosphorylation study, where we think that if you want a protein to be function, it should be a phosphorylated one or glycosylated or acetylated or any kind of other PTMs. Okay, usually it's considered a functional protein. Okay, it may be phosphorylated or it may be protein kinase. Okay, so it is like on and off, like switching off on and off buttons. Okay, so in a general term to understand. So what we did is like we usually work on, for example, if there is one gene, okay, so one from one particular cell, okay, the whole genome, there is 25,000 an average genes are there. Then we do the transcription studies. Okay, transcriptome is like almost 100,000. Okay, in India we call as like 1 lakh, right? I think so. Am I right? So one in India we tell as a 1 lakh. So it's usually called as like 100,000. Okay, in translation where the more than 100,000 proteins or proteomes are there, and then if you talk about like different modifications of that, you can able to get like 1 million at least. Okay, but when you're able to see the gene to the whole end of the protein, you have the huge complexity of that. For that one, we use like mass spectrometry based proteomics. Okay, which helps. Okay, which helps in like identification of like functional side. Okay. Okay, so now let's talk about this one. So what happened when we started to give the medicine? Okay, so when we started to give medicine, okay, so the present drug which we are prescribing right now, so it works if you give to 100 people, okay, it works only like in the case of depression, the drug which is currently right now in the market, okay, And then if someone is suffering from depression, okay, if someone is suffering from depression, so there is only 38% of the patient doesn't have any effect on the drug. Okay, when you talk about asthma, there are 40%. There is no effect. Okay, when about diabetes, arthritis, and Alzheimer, you have 40, 50, and 70. Okay, but in the case of like cancer, in because now there are too much variants and then too much mutations, are not actually okay works in some part, but it's work on like 5 to 10%. Okay, but we have alternative drugs as well, which able to, because based on the trial and error basis, so physicians or oncologists suggest some medicines, okay, where we have limitations for the targeted drugs. For example, as you all might have heard, okay, so if someone is suffering from fever, okay, if you're going to the doctor who is nearby to your locality, right, so like if 10 of your family member go for the like to the clinic, so they'll give a taxim shot, for example, okay, which is antibiotic, or give some antibiotic, the same antibiotic for the fever. Okay, so when once you see after a week, there'll be like almost 4 to 5 people will be cured completely with the drug. But if you see like 5 to 6 people, they have not get cured completely, right? Being Indian, what we tell is like, oh, this doctor is not fine, his medicine is not working. It's not like that. He give the medicine which is for that specific fever or cold or other things, okay, but it's actually doesn't suits you. Okay, so is it anybody here like this kind of scenarios? Yes, no?
Not from my side, sir.
So that means that if whoever is taking the medicine, they're completely cured. There's no one like something, oh, we go to this particular doctor. I mean, like it was not working. Yes, no?
Sir, actually, this
sir,
happened.
not really.
I mean,
Sorry?
now it's fine, but earlier it happened with my mother.
Okay, what happened is that?
Sir, actually, my mother has rheumatoid arthritis. And means this year and the last year, she has been visiting the CMC Vellore, and now she's, I mean, in control. But before that, in our company hospital or in various hospitals in Kolkata, the treatments given were not up to the mark. And I mean, she was not in a stable state after that also. After various treatments, nothing was cured. And so we had to go like this on the south treatment like that. So that is
okay. Okay, yeah. So we do work on like rheumatoid arthritis and other things, like for some time. Okay, so but depending on the patients how they respond and everything. So what we do is like we work on targeted treatment, like exactly, like doctor might have been prescribed one particular drug which is for the arthritis itself, but that is not suitable to your mother. Okay, so what we need to do is we need to diagnose specifically on her sample, okay, blood sample or other things, okay. Then we need to identify which exact drug will be suitable for her. Then we need to give that. So that is the thing called as like precision and personalized medicine. Okay, here you can able to tell like where we work on like truly on individualized medicine. Okay, so where we don't fit that one size can be fit for all. Okay, right? So in 2015, from US, from NIH, other people, we started research work. But truly, we started research work in 2013, or working on individualized approach. Okay, where we have like, because when you talk about like each genome makeup is very different, each lifestyle is different, and the treatment also should be a trialed one. Okay.
Sir.
So yeah, please go ahead.
Sir, in the previous slide, you just said the personalized medicine, but is it possible for everyone to just screen up all the genomic things and then starting the actual treatment? Isn't it very long time taking?
Okay, let me tell you, that's what we do right now in India. So even Ghazala is one of the main part of it. So we work on that one already. We are working for the past few years, but not much people are aware of it. Okay, we are giving individualized and we do the whole genome sequencing, targeted sequencing. We are doing it already, and we are helping so many different patients already who are getting complete cure. We work on like very different kind of thing. For example, the first thing is like kind of cancer, and second for diabetic, and for gastrointestinal studies, and then including some are related to the female studies like PCOD, PCOS. That is like also we do, but not for the genetic testing, but we do cure within 3 to 4 months on different kind of other things too, like hypertension, or some people have the cardiological like blockage in the heart, right, on the heart vessels, in particularly coronary arteries. So we help in like within 200 to 260 days to remove those arteries, to remove those plaques within the cell, I mean, within the coronary arteries, which maybe later on leads to myocardial infarction, in the general term called as like heart attack. Okay, so we are working on that already. But precision medicine for the whole genome analysis, sequencing a whole genome or every gene of a person, we do it already. Okay, so if you're interested to know more,
Uh,
okay, yeah, go ahead.
Another question is there. So if someone is in the last stage of his or her cancer, um, is it feasible to treat him using this personalized medicine approach?
Okay, let me tell you one thing. Okay, we are Indians. Okay, true Indians. So we go to hospital first. I'm talking about different stages of how we are. Okay, so what we do is like we don't go to hospital. Okay, one year is already done. Okay, so we have, doctor always advise we need to go for normal throat checkup, body checkup or blood check and everything, okay, for the hematological study and everything. But we don't go. We go only, okay, we go only when there is a issues or health problems or something. Am I right?
Yeah, actually, that's the real.
We never go for the general checkup just to check how is my health. Is it okay? Now we are like almost 50 to 60 people right now in the presentation, right? So is it anyone going yearly once to check their health conditions? Anyone regularly for the past 10 to 15 years?
No, sir.
Nobody, right? So because we people go only if there is something extreme, even if there is normal fever or slightly getting started, they think that okay, it's nothing. We can do this one and that one, that's fine. Why we had to go for diagnosis? And specifically the current situation we are in right now, okay? So in that one, nobody wanted to go anywhere. Even if you're suffering from fever, you don't want to get out, right? They think that it's corona or COVID, another thing, right? So they have a different kind of things in that. Understand?
Yeah, so
So in that
that's.
case, that's the first thing. I'll come to your question after that. Okay. So can we able to
Okay,
treat
okay.
a patient in the later stage? Okay. When the patient has already an issue or the pain in the body, then only they will go to the doctor. Okay. Then doctor will start to prescribe a generalized medicine, which he or she has studied. Right? Am I right? Yes, no,
Yes, sir. That
nothing.
is the main. Yeah.
Yes, sir.
Okay.
That
Yes,
is
sir.
the main thing.
So, can you point out any one doctor who is taking the research article which published last week and then giving the treatment based on that?
No, sir. Those type of doctors are really rare to be found out.
Hmm. But in my place, we do that. In our places, we do. Okay, where we take recent research publications and we do have our own research articles, our own patents and everything. We utilize those one and give the treatment. Okay, but already FDA approved drugs, we do. Okay, but which exact medicine that will be suitable, right? Understand? So I'll come to the question. So that let me answer the question. So what happened? Your question was, can we able to treat the patient who is in the last stage?
Yeah.
First, for example, okay, if the patient is already going late to the doctor and he is giving the medicine whatever he has studied, okay, already like 6 months has gone because the pain should be of little higher. Okay, then only they will go to doctor, and afterwards it start to progress faster. Then the doctor gives medicine. Okay, it affects or not. Okay, its affects or not, but they started to continue the treatment. Understand? And
Yes.
then what happened if they realize that medicine is not working at all? It's almost very late, right?
Sir.
That's the time most of the times the patients come to us for the whole genetic testings and everything, where we diagnose and then give the exact drug which is suitable for them. So eventually, most of the patients who come to the later stage itself with us, right?
Yes, sir.
Okay, so that's how. But yeah, we could able to treat. Again, but there is a condition applies. Okay, so first is based on the vital parameter of that particular patient. So if the patient's hemoglobin is too low, and the body weight and other things is where he don't have stamina to hold on the treatments, then we couldn't able to do. If the patient is healthy and hematological study and other things are really fine, where he can able to bear the pain even if it's in the later stage. Okay, where in the like,
Ready for
sorry.
Yeah, yeah. You can
Okay,
please.
someone is talking behind. Okay. Okay, so yeah, so what happened, like we do like individualized diagnosis, okay, using like precision like like diagnosis procedures with high throughput sequencing with the whole, and then we also give exact which medicine to give to the patient, and we give a separate report to the doctors who is taking care of that particular patient, and the patient write down that particular prescribed medicine that we give based on the latest research work and then genomic study, and then that medicine is exactly suitable to cure the patient. Okay, so that means that still even if it's later stage, we can able to cure. You got the answer?
Yeah.
I mean, like your explanations and everything, you understandable, right?
Yeah. Perfect.
Okay, great. So yeah, as we talk about like whole, if we have a genomic information, okay, which allow us to predict the, I mean, like what makes the I mean, like person, I mean, like how it developed, we can able to know. Okay, what the course of this disease we can able to know, and we can able to see how it respond to the treatment as well. Okay, with the genomic information. Okay, and now this is what I was talking about exactly. The current treatment is about if there is a cancer patient, for example, is suffering from colon cancer. Okay, so if we give the treatment, okay, if you give the treatment, so sometime what happen, it may be affect all the three people, good effect, and then it may be no effect at all. Okay, and there will be some patient who will be having very adverse effect, which is called as like side effect. Okay, so for example, if there are like, how many people are there? 7, right? And the completely treatment will be only for 3, right? And then 4 are like they don't know where to go, and one is almost like he's into serious side effect, right? This is the exact condition of the treatment right now, where one treatment is given to to all. Okay. So this is one of the latest advancement in the field of medicine, which combines latest research from like biotechnology, molecular biology, bioinformatics, and other things. Okay. But now what we are planning is that having a future medicine. But this is not future, we are currently doing right now. Okay, where if we have a patient, we take blood, DNA sequencing, and then we take our urine or even tissue biopsies. Okay, where we give, based on this one, we give specific medicine for the specific set of patients, and where you can able to see a very good effective. Okay, effects can be visible, right? Understandable?
Yes. Yes, sir. Yes, sir.
Any doubt in this two? Any doubt in these two slides?
No, sir. It's fine, sir.
It's clear and understandable, right? So this is the main thing that we can able to understand. Okay. And then, yeah. So here, let me go on this. So what we do is like we take the we usually check for the biomarker study, okay, where we get the biopsy samples and then we identify the mutations and we get the results and we see what exactly the drug, like here there's a drug, okay, drug can be given, okay, this is how we give the treatment. And really biomarkers are helping for the personalized cancer care, or even it can be from the KRAS, okay, it can be from like ALK, EGFR, okay, and specifically for this is the example which is given is for like small cell lung cancers, okay. So when you talk about the small cell lung cancers, okay, so here here this all the genes are there, which is like example I have given here, okay. Apart from that, there are more than 105 genes are there which we usually target for the cancer, okay. And then after that, like this is one of like therapies. What happens is that like after we give the therapies, for example, let me distribute on 3 different aspects. Some people are there, they respond, some people they don't respond, okay. So we take that one as a different biomarkers, okay. So if I, for example, biomarker 1, biomarker 2 for the same disease, okay, for the same disease we can able to have, say the different patient can have different biomarkers, so we can able to go for like different therapies by characterizing them, okay, based on cells, based on like cellular study, based on like molecular study, okay, and then like based on bioinformatics study which Ghazala is working on, okay. And then if some people are there who don't respond, so we can go for new therapy in the clinical trials, okay. So this is like the same, we will not be working only on the like which medicine to give, we also work on like how much the dosage that we can able to give. For example, even the dosage is of really important, okay. So this kind of, which kind of dosage works and how high level of like the some patients effects, okay. So for example, some patients need like high amount of dosage, some patients need like low amount of dosage, okay. Some are like different drugs even. So earlier we have were talking about like which drug to give, which is on the precision drug, which is on the precision, which is on the precision one, okay. Then afterward we have the standard dose, we have lower dose, and then higher dose as well, okay. So you guys understand? So earlier we were talking about which exact medicine to give, then we were talking about like how much dosage we can able to prescribe. Are you getting my point? Hello?
Yes, sir.
Okay.
Yes, sir.
So in this one,
Yes,
in
sir.
this one, let me come to the biotechnology. I mean, like cellular. I mean, like I hope you guys know what is cell line studies and everything, right? Cell line.
Yeah.
So anybody know what is cell line? how we do cell line culture and everything? Any idea? Hello?
Sir, about cell line.
Yeah, cell line studies.
Yes, sir. Mainly, we cultivate or we grow a certain type of cell, and then we use the cells for regenerating again and again and again, and then a cell line is formed.
Okay.
Multiple culturing, subculturing. By this method, we can achieve a cell line.
Yeah. So during the time, what we do is we take the biopsy sample. Okay. So we take the biopsy sample of the cancer patient. Okay. Then we see like what exactly to be given. So like, for example, once we know that what is the targeted drug to be given, okay. After we take the target drug, we need to identify how much dosage is to be given. Because when we give to the physicians, okay, so that time we need to prescribe, we need to mention the dosage as well. Okay, so in that case, we use the cell line study, okay, where we grow the cancer cells from the patient. Then we work on like giving the different dosages in the cell line, different kind of the same cells we grow in different medium, different plates. Then we give the different dosage and see which one actually works well. Sometimes what happens when if you give the lower one, it will be started to like affect slowly, okay, or it will be taking too much time. Sometimes what happens if you give the higher dosage, it can be or it may be faster and it has some adverse effect as well. But what we do is like we need to maintain a normal level which can go in a good way with the timing and the dosage as well. So that like patient will not be suffering from other things with our study. Okay. Understand?
Yes, sir. So,
Is it clear?
sorry.
Yeah, go ahead.
Sir, I wanted to ask a question. When we work on cell lines, we have modified the conditions of it. But when cells are present in the body, the conditions are very different. So how is it feasible that we are working
No,
on a
we can grow the cells in the same condition.
Yes, sir, but in the body there are very other factors that affect which we cannot.
No, we take exactly cancer cells alone. We take the biopsy sample from the cancer cells alone.
Okay, sir.
The tissue biopsy which we get, it's from the cancer cells, and we also get from the normal as well. So we grow.
Yes, sir, but in the body there are various factors.
Your voice is very low.
Lactate accumulation and various other factors. So how do we?
Hello, your voice is very low. I couldn't able to hear you.
So, sir, yes, sir. I was saying that in the body there are various other factors like lactate accumulation and various other enzymes that affect the cells. So, how do we maintain this in the cell culture? And if we do so,
that's a biochemical study. That's a different thing. Like enzymology as things. That's a different thing. Here we talk about like pharmacological aspect of that. Okay.
Yes,
because
sir,
pharmacological aspect, so it's already studied. You're talking about this thing like other enzymes, other things will be involved. How we are going to work on that one. So actually, when the drug is patented, so during the clinical trials and everything, those things is already studied. How it has been effective, how the pathway will be effective, right?
Yes, sir. Yes, sir.
If that is not studied, then the drug will not be approved at all. Okay. So that was already done. Okay.
Yes, sir.
Do you understand?
Yes, sir.
Hello. Yeah. So but if someone is interested in that one, we can able to again work on that one on the biochemical aspect of that. Okay. But without that one, the drug is not approved. So we are taking mostly approved drugs, and then some drugs in some conditions we are taking on a under clinical trial drugs, which if it shows effective. Okay. Even in that also we have already the report that how each enzymes works with this particular drug and everything on the cells. Okay. Hello.
Okay, sir. Thank you.
Yeah. Okay, so here is the same thing, like how we, what are the like molecular profiling that we do? Okay, so for the cell line structures is there, and then like how genotyping we do, sequencing for the Shangar for the confirmation of the mutation, and then we check for the biomarker for the predictive sensitivity and specificity of the drug. Okay, and we also analyze like how we go for the adverse, how we can avoid the adverse effect or side effect of that. And for that one, there are already many different companies. And then the most importantly is that, as someone was talking about, like it's how it's possible to sequence the whole genome or the whole gene. So is it been practically possible? So I think like everyone knows about what is Human Genome Project, right? Right?
Yes, sir.
Okay.
Yes,
So
sir.
what is
Yes, sir.
other Human Genome Project is from company, from the government. Okay, there is one company which also worked with together with the Human Genome Project. Now with the Human Genome Project, there are two different. Okay, one from the government funded one. Okay, second is from the commercial company. So do anybody know what is the name of the company who sequenced the whole genome? Any idea?
No, sir. We don't know.
Okay. So you're telling no, some people are Googling. Wait.
Sir, was
Okay,
it
I'll
the
tell you.
welcome blast?
It's a Sclera Biotech. It's Sclera Biotech, which works on which help together with the whole genome project, Human Genome Projects, on a commercial aspect. Okay. So what happened? It was a few billion when first human genome was sequenced. Okay. Now recently we started to reduce the cost and we improvising the technology so that it can be utilized by any person who is suffering from cancer or any other diseases. Okay. So now we could able to make a whole genome of a person. Even if you guys wanted to know your whole genome of you, if you want to have a whole sequence of your own, you can spend around like minimum of like from $1,000, which is around like 760 to 70,000 rupees, Indian rupees, right? But it depending on which kind of sequencing that you do. Okay, this is not like personalized genomics, where you can able to sequence your own genome. Okay, that we usually do with by ourselves itself. Okay, and then there are some companies which also offers, okay, apart from us, which is like for profit like organization, a genomic company which is called like 23andMe, okay, Nevigenex, and then Decode Me, and other people are there. Even there are few in India, and we also work together with them, okay, where we sequence the whole genome of a normal person or even a disease condition as well. Okay, where we are able to identify like for the prostate cancer or even like Alzheimer diseases or myocardial infarctions, okay, or other syndromes as well, which are of like genetical disorders. Okay, and then we do have published papers in cancer cells, okay, which is one of the highly impact factor journal in around all the publication in the world. Okay, so where we worked on like human early onset of gastric cancer, okay, among like patient less than like 35 years old, okay, where we took like normal, like you can able to see, we took like almost a young patient with a diffuse type of gastric cancer, where we got like tumor normal from each patient. T is a tumor tissue and N is adjacent normal tissue. And we did the whole exome analysis, and then we did whole RNA sequencing analysis, and then we work on like whole proteome level as well. So this is the kind of like, you know, I think everybody know what is central dogma of biology, where like, right?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, that's great. So DNA, RNA, and then protein, right? So we work on DNA, RNA, and protein level. Okay, so to identify also how the functions and everything happens, we did work on like PTMs, Okay. Um, Okay, so then afterwards we analyze few things where the mutated population correlation, where we have analyzed also in the signaling pathway level. Okay, we correlated mRNA and protein expression level, and then check it out how the patient survivability. Okay, how long the patient can be survived. Okay, we have our own data which supports those things. Okay, we also identify like how the subtypes of the cancers and everything characterized. Okay, and then like we also prove that one with the cellular signaling pathways of those phosphorylated proteins. Okay, and then these are like few technologies which I was leading as the author. Okay, like few are published in like in analytical chemistry paper, on mass spectrometry paper, and then few are in Cell and other journals. Okay, in Nature as well there are. Okay, and this is the group which works together with me, and okay, Ghazala is also there, one of the prime member in the group. Okay, with me. So these people are there who work as a like mentor. Okay, so whose name is Sunwong Lee and Richard Dick Smith and Bin Zhang, and then Kim is there, and Professor Park is there. Okay, and apart from that, and these people are like kind of office people who work together. And apart from that, primarily Ghazala also works. Okay, and then Mahmouda, another few students are there who joined as an intern, or they joined as as for their master project students, and they again started to continue after getting interest in our study. Okay, so they still continue working for a few years right now, and few will be getting a job in my own company as well soon. Okay, and then we as in my own hospital, we work on like personalized treatment for the prostate cancer, where we claim to be of giving 99 to 100% of the treatment for the prostate cancer to get completely cured. Okay, and we started to work on like stomach and gastric cancer to help on, and we do work on like tongue cancer right now. This all the patients are there right now who is undergoing treatment, who are responding very well. If someone is interested, okay, if they're like, oh, I wanted to work on like tongue cancer, I'm interested in that, so we'll be happy to give you a chance to learn, study, understand. Okay, and if you are interested, you can able to do your projects or some other things. Okay, that depending on your interest. Okay, so if you're interested, you can feel free to contact Ghazala. Okay, or me for that one if you are interested, and you can able to have a talk first with your faculty member, discuss with them. Okay, so through them you can easily contact us. Okay, and we'll be really happy to do research work, and we'll be collaborating with your faculty member with some students. Okay, so that could be of really fruitful, because there was some questions about like how you in the questions that you have asked in the group, right? So can we able to do some research projects or something by sitting in home and everything? It's really is possible. Okay, for that one, I could recommend first you discuss with your concerned faculty member. Okay, then you can email us personally as well. Okay, so you can able to have like email address which I can able to give. 1 second. So so we have an email address here. Okay, I can make it a little bigger for you. So if you wanted to have.
Nice. It was very nice opportunity for the students.
Yeah.
Riddhiman.
Yes, ma'am.
Now please open for the discussion.
Yes, ma'am. Actually, sir, can you are you sharing your email ID?
Oh yeah.
So you told yes. So
this is my team email ID. So anyone from my team, they will be like, if you have any query, or if you wanted to have some projects, if you have idea, if you're interested in our work or something, yeah. But first I could recommend to have a discussion with the faculty members. Okay,
definitely, sir.
Yeah, please go ahead with the queries.
Sir, yeah, there is one question from the YouTube chat. So Ajay Shinde is saying, how does methylation lead to cancer A-level biology?
Cancer? Which one?
A-level biology. How does methylation lead to cancer A-level biology? It was the question.
So it also like it's as I told, like when you talk about like okay, when you talk about the molecular level of it, okay, so there is not only one particular PTMs like methylation, okay, there are few conditions are there, okay, in a particular metabolic pathway or biochemical pathways, okay, in that specific protein, okay, if there is a methylated, sometime what happen, it's may normal methylated one, so which may not be causing cancer, but you can able to see that one as a result, okay, in your data, if you sequence the data, so you can able to see those methylated one, okay. But what happened in one of the study which I published like 2 years before, so where we identify three different kind of PTMs, okay, where the mutation was there present, okay, in a specific protein, okay, and we have seen that particular mutation in the DNA and RNA level, okay. So where one methylated has been shown as a functionally active, and it can be a biomarker, okay. When you talk about like PTM level, for example, it may be acetylation or methylation or phosphorylation, glycosylation, but predominantly PTMs are the first one is always like phosphate group, then glyco group will be there, and acetylation and other things, okay. So but when you talk about like functionally active or something, where phosphate groups are like predominantly studied, where we have like good evidence to work towards that, okay. Not specifically because like only methylation can be in a normal one, okay, but some other things should also will be there in a molecular level changes, then it has some role, okay. So then we can able to study that one separately on a methylation studies, okay. Then we need to see the role in a specific pathway, and we need to see which specific protein actually it's methylated, okay. So based on that one, it is actually involved in like carcinogenic pathways or oncoproteomic pathways, okay. So then that is like kind of an interest that can be a good biomarker to study, or we can able to see like how that can be like a linkage or progression in cancer, okay.
Sir, Orko Prabhu here, sir.
Yes, sorry. Yes.
Sir, I just wanted to know, sir, how helpful radiation and chemotherapy are in curing cancer?
Yeah, it's really helpful. It's really helpful. But again, like chemotherapy is really important. That's a common thing that we usually do for treatment of the cancer. Okay.
Yes, but so in chemotherapy, there are a lot of side effects.
So yeah, side effect we have. There is side effects, but for that one instance, recently for more like 4 years before, we started a different kind. We started to prescribe some medicine only for the chemotherapy patient, where they will be having less side effect. Where we use as a chemo detox, where the toxins which is there inside a patient's system, it can be removed or detoxification can be there.
Okay, so actually I was saying because some years back, about 4 or 5 years back, my uncle had prostate cancer. He's cured now, but because of the chemotherapy, his bones are extremely brittle, to the point that the doctor has told that he cannot fall down. If he falls down by accident, he might break his bones.
Yeah, that is a common thing. It's yeah, that's a thing that we can able to see. But there are some other things for that. Okay, to strengthen the bones and other things. So still, that is also there are some treatments. You can same consult the same doctor. Okay, or even we will be happy to help and give you some suggestions on like what are the things that we could able to do, because we have seen few patients. I mean, like who has an issue like for the longer term. Okay, due to the chemotherapy. So what we have to do is we need to have a chemotherapy as well, but Apart from that one, we do need to have some external support as well, like for example boosting for the immunity, and then like we need to have a detox as well, so that like we'll not be getting too much side effects due to that. Okay, so which is like kind of slowly it's getting into normal. Normally people are coming forward to have that one, like whoever is like, for example, as you guys are getting to know few things right now in this session, right? So like you guys will be having an idea, okay, this all the things exist actually, that we can able to go ahead and do those things. Like for example, not only when the chemotherapy is going on, we have a something called as chemo detox as well, which can surely remove the toxins, okay, from the patient system, and then which will not have much adverse effect. Okay, it may be even due to chemotherapy or even radiotherapy. Okay.
Okay, sir.
So I'm having a question. As you said
Yes,
that
please go ahead.
mobile radiations cannot cause cancer, can it accelerate the
Yeah.
growth of already existing cancerous cell within the body?
No, it's not. But there is no proven itself for with that. Okay, it's like for example, everybody knows that one. Like if you're too much exposed to the radiation, for example, like, the working who is radiologist, who is working on a X-ray department, right? So there are some, okay, but it is of like, for example, if because like when compared to the from mobile phone, okay, if it's truly causing cancer and it is really progressing too high, then the mobile phone is banned, right?
Yes, sir.
Yeah. So, but even in the case of like, but the intensity of different. Where in the case of like in X-ray emissions, okay, the intensity of the radiation is very high. Okay, if for example, let's take it like 1,000. Even if you wanted to calculate with the mobile phone itself, if 1,000 to 10,000 is a level of radiation if you get from the X-ray machines. Okay, in the mobile phone or other things is a permeable, which is like almost 2 to 3% if we do study on that. But still, there is no other, there is no like evidence that it has a role in developing a cancer or causing a cancer or improving the cancer who is already suffering from it. Okay,
Yes, sir.
because there are some people who did research work on that one, still they couldn't able to find an exact what to tell like evidence towards those hypotheses, so it's become as a myth. Okay.
Yes, sir.
Yeah, great.
Sir, actually, Shantika, please wait. So, sir, there is one question from the chat that they couldn't see your mail that you shared. So, can you please
See my
do it
name?
once again? See your mail that you say that you shared, sir, few times back.
Okay.
So can you please do it once again for them?
Need this one? This email
Your
ID you
screen
mean?
sharing is off.
Yes, sir. Your screen sharing is off.
So someone can type, oh, the email ID is onco inam, ONCO INAM at the rate of Gmail. Or someone can type in the group chat.
All right, sir.
Okay. Yes, the next questions, please.
Yes, sir. I was just talking about the patient. If we think the drug analysis or new therapy searching differed from the patient's aspect, if we think that the new drugs that is in the market or in the clinical trial phases, and those are actually going through several steps and finally coming into the market for the patients. But when we are talking about personalized medicine, it needs specific dose, specific types of drugs that should be provided to the particular patient, but is that available in the market ready for
Yeah. So in that
patients?
case, what happened? So we usually use, what we do is like some drugs are there. So which is like under trials. Okay. So we will know what exactly biomolecules, what is the molecule which is present there, and based on that one, we'll be having a generic name and everything. Okay, with a different additional like side chains attached to that, which makes them like more active or more effective. So what in that case, what we do is, for example, if one drug is there which has been identified. So in our study, what we do is like we usually go at the first for FDA approved drugs, which is already available in the market. There are thousands of drugs. Okay. So if not, we'll go for FDA approval, which is under clinical trials. So in that one, we identify some drugs which may or may not be in the market. Obviously, it may not be in the market. But what we do is we take the chemical structure of that particular drug, we search for analog of that one in a other drug which is available. Okay, for example, let me tell you one story. So a few years before, what happened, we got one mutation. Okay, so as everybody knows, like mutation happens in the, so one patient who is suffering from cervical cancer. Okay, and then what happened, we were checking for like so many different drugs and we couldn't able to identify. It was so difficult and frustrating. Okay, then what happened, when we were analyzing the data, okay, we were also prescribing few medicine which is related to cervical cancer. Okay, then we took all the drugs related to chemotherapy or like cancer related drugs. When we identify the mutational data from the patient and the drug, so we got the mutation exactly, which is from the BRCA, which is responsible to initiate and progress breast cancer. But the patient has a mutation in BRCA, and this patient is suffering from cervical. So when then we started to draft a pathway. So how this particular mutation, which usually causes in breast cancer, how this has been causing in this particular patient. So that was one of the very interesting like study that we did. Then what happened? We started to give breast cancer related drugs to the cervical patient because of the mutation, and then we also give few additional drugs, okay, based on her vital parameters. Then she started to shows a very good result. Understand? So one we take under clinical trials for the analog, or we give other similar kind of drug from the approved one, or we combine two to multi drugs to target to help the patient to get cure. Okay. So, but still, when we have the drug which is under clinical trials, we have few limitations. And sometimes what happen, prescribing that specific drug which is under clinical trial may not be fine. It is not sometimes illegal even. Okay. So we use the analog of that one, which is already approved. And then we added some other drugs together. Okay. Not single drug, multi drug, and then we get to help the patient to get cure. Okay.
Yeah. So the patient, each and every patients are treated like several trials. If none of these are like standard or anything, they all those are personalized as well as experiment, right?
No. Recording the drugs one, right? For the get the approval and everything, there's a completely different study. Okay, where we have a different 4 to 5 clinical trials methods which people usually follow. And that's how it should be when you send the drug which you have patented to the FDA. It may be Indian FDA or it may be from US FDA. So there are some timelines and there are some procedures starting from which chemical molecule. For example, if you talk about the drug, there are some rules and regulations. First, all the drugs, which is allopathic drug, for example, okay, it's of some kind of chemical and biomolecule or enzyme. Okay, it may or may not have side effects as well. We need to check, and we have separate parameters. We have we can go for cell line study, in vitro, in vivo, in silico. All these things we need to submit. Then after biochemical things, okay, and then toxicological study. You know ADME-Tox, right? So in ADME-Tox and other things, everything we need to provide. Then it will go. And still after that, after submission, so FDA, they by themselves they have a team where they study again all the things. It takes almost 5 to 10 years to confirm about the drug. Okay.
So mainly, sir, because of the time constraints and other events are also scheduled up right now next. So I think we will have to sum it up here. So we at Team SABBIT once again sincerely thank Ms. Ghazala and Dr. Inam for their informative session, and also for sharing their expertise with the participants. We are definitely interested to learn and work more regarding the works you have shared. So once again, thank you so much, sir, and thank you, ma'am.
Yeah,
as well.
welcome. Yeah, it was nice talking to you all. Yeah, thank you so much. Thank you for having us, me and
Our
Ghazala.
pleasure, sir.
It was really nice having a discussion with you all. Yeah, thank you. Okay, yeah. Take care. Yeah.
Okay, so
bye.
Thank you so much. Have a good day, sir.
Thank you so much, sir.
Yeah. Bye.
So the first session for day one has come to an end. The feedback form has been provided in the chat box for both YouTube as well as Gmeet, and you need to fill up the feedback form for both the 2 days in order to receive the certificate. So after a short lunch break, we shall commence our day one second half featuring the debate competition. The event would start from 2:30 p.m. sharp, and the detailed description about the event shall be discussed at that time. So see you all at 2:30 p.m. and thank you so much everyone for attending. Thank you.